Good morning. This is Thursday, July 23rd. Any questions before we get started today? Great question. I posted the grades. I got them. I finished the grading late Tuesday night, so I did get the grades posted, but I have not released the questions yet. So I will be doing that just like that last time. So they will be today. Thank you for reminding me of that. So again, I will release the exams for review after class today. And then they will be available only until class on Friday. So you've got about 24 hours to review your exam. If there's any questions or anything else that you have, I purposely did not release them yesterday because I wanted to make sure that I could announce it to the class when we did that. So I know as soon as I got it graded, I wanted you guys to be able to have your grades, but I just wanted to make sure that that you guys are aware that when I'm releasing it, because that's the whole point. The whole point is for you to learn from what you see on there and what you got. So definitely we will be doing that today. And actually, I'm going to myself a note for that too, because once this is gone, I will forget it. All right, excellent. Great question. Any others before we get started? All right, as crazy as it seems, this is actually our last Thursday lecture. So that's kind of nuts. I still feel like we're just getting our legs under us in this class, but that's kind of summer school for us. So this is our last Thursday lecture. We are going to, like we did actually mentioned last class, we'll finish with the anatomy of the brain, finish with our lab first, then switch back into lecture mode, and then hopefully get to the cranial nerves today. We should be able to at least start it today. If we don't finish it today, then we'll finish at the beginning of class tomorrow. You still have a couple more assignments due, 4 more assignments due. Tomorrow, your physio exercise 3 is due. There are 9 activities for that. Again, they're not hard, so just time consuming. So hopefully you've gotten started on that. Otherwise, you have a busy night ahead of you tonight. Monday, your 15-point nervous review is due, and that will be graded for correctness. So you do have that. And again, today's information, if you haven't started on that yet, that's actually a good thing. Today's information in today's lecture, when we finally get a chance to talk about the spinal cord, should hopefully be the last bit of information you need to help you to be successful on that. Tuesday, 2 more assignments due. Unit 14 review is due. And again, the reflexes home lab is due. You're doing procedures 2 on the pages 371 through 374 and procedure 2 on page 380. Okay, so the goal of those are for, again, you have an opportunity to, these are things we would normally do in the classroom, but you get a chance to play with these things at home. Most of them are things you should be able to do at home. Anything you can't do at home, do as a thought experiment. And then it's all tests. Thursday the 30th is the lab and lecture exam, and you guys know what to expect from the format of that. You've seen that, you know what to expect from that. So that should be hopefully successful for you. And then Friday, we have the final exam. The final exam is 100 multiple choice questions. You'll have 100 minutes to complete it. And again, like our lab and lecture exams, it must be completed during the class time. So between, it'll become available at 8. And even if you start at 12:30, at 12:35, it shuts down. So again, you must complete it during that time. So plan accordingly for that. All right. Questions on any of that? All righty then. Let's go ahead. And again, that final exam is cumulative. Everything we've covered in this class, pretty evenly distributed over the material that we've covered. All righty, let's go ahead then and switch back into lab mode. All right, we left off last time and we had been working our way through our lists of the brain anatomy, and we've done most of the heavy lifting on this list, but we ran out of time, and so we didn't get the opportunity to finish it. We had talked about the telencephalon and all of the structures associated with that, both the superficial ones in the cortex and the nuclei deep and some other white matter structures. We have talked about the diencephalon, the thalamus and the hypothalamus, and the other structures that surround them as well. And so what that brings us to now is the three M cephalons, mes, met, and encephalons that we need to talk about. And again, as we mentioned, the dividing point for those are basically this small narrow region there is the encephalon, or what is also sometimes referred to as the midbrain. The two enlargements, the pons out here and the cerebellum out there, is the metencephalon. And then the inferiormost part of the brainstem that forms the medulla oblongata is the encephalon. In the same way the telencephalon is the cerebrum, and the cerebrum is the telencephalon, the encephalon is the medulla oblongata. The medulla oblongata is the encephalon. So those two terms are kind of mean the exact same thing. They're interchangeable basically in those senses. We saw another view of these as we flipped it this way. This again is an anterior view. And here in green, we see that narrow region that is the mesencephalon, the mesencephalon. And here from the front, we see the most prominent structure of the mesencephalon. The most prominent structure of the mesencephalon are these big, huge, knuckle-like prominent projections that stick out, known as the cerebral peduncles. A peduncle is basically just a fancy name for an enlarged white matter tract. These enlarged white matter tracts go to the cerebrum, and as you can see, most of them go to the thalamus first, which remember we said was the relay station for the cerebrum. And so from the anterior view, they are the most prominent feature. If we actually cheat and go back a picture, we see them here as well. This big, large, prominent white matter structure we see to the front is primarily that cerebral peduncle. So we do have that there. So we can see it from that view very nicely. We see it from this view very particularly. However, the other way we can see it is if we were to actually take a cross section through it. If we were taking a cross section through it, and notice in this case, we are looking at it from the posterior side. So this is posterior and this is anterior. Again, as we mentioned, this large prominent white matter structure right here, sticking out like a big knuckle, are these cerebral peduncles. And like I said, they're basically just white matter carrying information up to the thalamus, and then from the thalamus to the cerebrum. All right. But notice as we look at this, we do see some other structures in here as well. Starting first with this hole, I know we haven't talked too much about the spaces inside of the brain and brainstem yet. We've hinted at them. We know they're going to be filled with cerebral spinal fluid. We saw the lateral ventricles and talked about the third ventricle in the last case. And actually right here is the fourth ventricle, this diamond-shaped region located here on the back of the brainstem. Well, what we have right here is a passageway that goes from the third ventricle, which is actually this space up here, and tunnels through the mesencephalon to get to the fourth ventricle. This space is what is known as the aqueduct of the midbrain, or what is also known as the cerebral aqueduct. Again, remember one of the keys to all of these are they're all spaces, and that is how I will ask you to identify them on the exam. And remember, these spaces are all going to be filled with cerebral spinal fluid. Cerebro spinal fluid, all one word. There are some additional structures here that we need to be able to identify, though, as we look at this. The first one of these, as you can see kind of nicely in this picture, and then I've got an illustration that shows it as well, but let's start here. If you notice, and actually, no, I'm not going to highlight it. I'm going to surround it. So let's use yellow. As we mentioned, the cerebral peduncle is white matter. It's axons, because it's passageways, information being carried up to the brain. Well, notice right behind it is this very dark stained region. This dark stained region is what is known as the substantia nigra, right? Because it is a dark substance. When it was first identified, it was a very darkly pigmented region, and so that's where it got its name. And it turns out what causes that dark pigmentation is our friend dopamine. Remember, we talked about dopamine is an important neurotransmitter, and we also talked about the important role it plays in Parkinson's. In fact, a diagnosis of Parkinson's can only be confirmed by a dissection of this region of an individual's brain. Because what happens is when someone has Parkinson's disease, the cells in this substantia nigra that produce the dopamine aren't producing dopamine. Remember, we said that was one of the symptoms associated with Parkinson's. And so this area is completely clear in color. It does not have any pigmentation. It does not have any color to it in this region at all. So again, obviously, having your brainstem dissected is not something that usually someone has when they're alive. So typically a diagnosis of Parkinson's isn't actually fully confirmed until after someone actually dies. We see it nicely here on the actual histology of this, but what we have here is this pretty picture. Now, again, notice, don't get confused. They've turned the picture upside down on us. So now anterior is down and posterior is up. I apologize. I have not had my coffee this morning. But again, we see those two areas we just talked about. Here are those big knuckles that are the cerebral peduncles. Here is that dark stained region known as the substantia nigra. But what's nice about this is this illustration allows us to see a couple other things. The first is this area right here. Notice just posterior to the substantia nigra, a dark stained region, is the red nucleus, called the red nucleus because it is red in color. But the red nucleus isn't on your list, so we don't really care about that. What we do care about is this shaded region that is just lateral to the cerebral aqueduct. This lateral region is a structure known as the reticular formation. And what this reticular formation does is it plays a very important role in our overall alertness. Right? What do I mean by that? Well, what I mean by that is my wife is a perfect example of this. She sleeps incredibly soundly with sirens are going off and dogs are barking, right? Gunfire is going off in the background. All those types of things can be going on and she sleeps soundly. However, when that newborn baby just whimpers just the tiniest bit, mom is instantly awake and alert and aware, right? Or when she hears the floorboard creak as I'm trying to get out of the bed to go play Xbox, right? She is instantly awake as a result of that, right? Obviously, what's the difference there? It's not the volume of sound. It's not the amplitude of the noise, right? What's difference there is the significance, the importance of that information. Different types of sounds have different significances and different implications to us. And as such, we have a different alertness to them. Another way that you may think of this area, this reticular formation, is another area that does get input from the visual system. So one of the things that happens is light, especially flashing light, like watching TV or things along those lines, have the ability to increase the activity in our reticular formation, making sleep hard. So one of the things that if you're having trouble sleeping, staring at your phone and the bright light from that or watching TV often can exacerbate the problem rather than helping you to fall asleep as a result of that because of that stimulus that is coming into this area. So this reticular formation plays an important role in our level of alertness and awareness. And like I said, different sounds, different stimuli are going to have different impacts to us and for different people as well. All right. Questions on that? Right, again, babies are a great example of this, especially if they have siblings, right? When if you, what can often happen is if you have the two kids in the same room, The baby's screaming its head off. And like I said, even just the whimpering of the baby is enough to wake up mom, but the baby can be screaming its head off and the sibling sleeps through it without any problem at all, right? Because the parents care about the crying, the sibling doesn't. So those are the types of things that we're talking about. It's not the amplitude, it's the importance of that sound that causes the significance. All right, questions on that? All right, I want you to notice one other thing, or really two other things, or really four other things. Let's start here with these two things. Notice on the posterior side, there are these two enlargements on the back. Notice collectively they are known as the superior colliculus. But let's change our view a little bit. I'm going to sneak back to the previous picture. Notice here we have those same two enlargements on the posterior side here as well. But now that we look more closely, we see that those are not one, not two, not three, but four. There are four distinct enlargements located on the posterior side of the brainstem. Again, when we take a cross section through it, we just see two of them, 1 and 2. Notice also, go back one more picture, two more pictures. When we look at a sagittal section, we just see two of them as well, 1 and 2. As we've seen, this one up here is what is known as the superior colliculus. This is the one that is the superior colliculus. And if that is the superior colliculus, what does that make this one here?
Inferior.
Inferior colliculus, absolutely. All right, so here we see them side by side, or really one above the other. So we can see the two different ones. Obviously, from the anterior view, we don't see them at all. We do from the posterior view, but it's got the section through it, so we'll just focus on seeing the superior colliculi, the two, plural being colliculi, that we see here, the two superior colliculi there. But here, we get that nice view of all four of these structures together. Now notice they've made it a little bit transparent here to show us the relationship of where that substantia nigra is, where the red nucleus is, those things that are deep to it inside. But what I really want to focus on, and again, I like this picture a little bit better because it's a little less confusing, that there are indeed four structures, two superior colliculi and two inferior colliculi. Four things total. Both of them have similar names, superior colliculi, inferior colliculi. So, since these four things are clearly related to each other, these four things collectively are given a name as well. Any idea what that name of these four things collectively together might be? If only I had a list of terms in front of me that I could read off of. There you go. The corpora quadramina, right? Quad means four. So the corpora quadramina collectively are these four things. Individually, we have two superior colliculi and two inferior colliculi. All right. That is the anatomy. What we need to talk about then is their function. Both, or let's say it this way, the entire corpora quadramina is about coordinating information. We take so much about our understanding of the world for granted, right? I can turn around, I can see my glass of water, I can pick up my glass of water, I can take a sip of it, and I can put it back. The ability to do that is an amazing, remarkable ability that we totally take for granted, right? We do because we think, well, obviously I see something, and where I see it, it is where it is in space. But if you think about it, that's not really how sight works. We haven't talked about vision yet, but vision works very similar to a camera. In ancient times, there were these things called cameras. Cameras are not the small devices you keep in your pocket that you use to text and to send emails and phones and all, I mean, all those types of things as well. Cameras were big boxes that had lenses on the front and film on the back, and light would come in and reflect on that light sensitive paper, and then it would be developed and turned into a photograph. And that's kind of how our eye works. Light comes into our eye and hits a point of our retina. And so your thought would be, okay, well, when light hits a certain spot on my retina, that tells it me where it is in space. But it really doesn't, because the problem is my eyes move. So right now, as I look directly into the camera, right, there is light hitting a port in my eye. But if I turn my eye to the right, or I turn my eye to the left, or I put my eye up, or I put my eye down, that same light that's coming in hits a different part of my retina. And it's even worse than that, because not only can I move my eyes, but I can move my head. So I can move my eyes to the right, but my head to the left, and then my eyes are the same place it was before. What we need to be able to do then, to really know where something is in space, we have to be able to put together two pieces of information, really two types of information. I need to know where light is hitting my retina and where my head and eye is in space. Or let's make that easier, where my body is in space. And I need to take those two pieces of information and I have to put those two pieces of information together. And that is what the superior colliculus does. The superior colliculus coordinates visual information and body information, where our body is in space. And what we call that is proprioception. There's a fancy word for that, proprioception, right? For those of you who, whoops, that's not where I wanted to put that. I meant to put that at the end. So I don't know if I can cut that. Yay, that worked. There we go. Where my body is in space, right? I can lean back in my chair. I can close my eyes. And even with my eyes closed, I am able to move my fingers to touch my nose, right? If I've had a little too much vodka, this becomes a little harder to do, but I'm only on my second cup this morning, so it's not so bad yet. And I am able to do that. That ability to know where your body is in space is proprioception. So basically, what our superior colliculus does is it coordinates vision and proprioception, puts those two things together, and that's what allows me to tell where my body is, look and see where that water is, and then reach out and grab that water. All right, questions on that? All right, then what we need to talk about is what it is that the inferior colliculus does. Someone's making breakfast, so I'll go ahead and mute that. Our inferior colliculus, at least I hope that's breakfast. Our inferior colliculus does a similar thing in that it basically coordinates proprioception, where our body is in space, and audition, hearing. Because the same thing. Now, granted, unlike my dog, unlike our cats, I can't move my ears around in the same way that I can move my eyes around. But as I move my head through space, as I move my body through space, is where my ear in the world vary? Does it change? Absolutely. So again, if I'm going to understand where a sound is coming from, I need to be able to coordinate that ability to know where my body is in space and where I'm hearing that sound so that I can turn to it. And again, you guys probably haven't thought of this in these terms, but you're aware of that. Because while you were sitting here calmly, and again, if we were in the classroom, this would be much easier. Because if we were in the classroom, I would have talked to whoever I sat in the very back of the class to let them know right now by the pull of my ear that what they should do is take their textbook and drop it on the ground. And if someone in the back of the classroom were to take their textbook and drop it really loud onto the ground, what would we all do?
Jump up scared, turn around.
Okay, well, yeah, sure, you might jump as a result to it, but you would also, the other thing you would do is you would, like you said, you would turn around. You would take every part of your body, your eyes, your ears, and you would point it all at where that sound came from so that you could assess if that was, something that needed to be a concern. We could assess the threat level of whatever that was, figure out and determine what that was. That ability to take all of our senses and focus them at one location, where our body is in space, where our eyes are looking, where our ears are pointed, all of those things, and point it at that disturbance to see and evaluate it. That's what we would all do as a result of it. Yeah, some of us would jump. I'd probably pee a little bit, but we would all focus all of our senses in that location to assess the threat. And that is what our corpora quadramina does. It coordinates those information of what's coming in and where our body is in space. All right. Questions on that? All right, excellent. Well, just that easily, we are done with the mesencephalon. So the mesencephalon was pretty simple, pretty straightforward. That was pretty nice. Excellent. Said we did most of the heavy lifting. Let's talk then about the metencephalon and the two things that go along with that. Notice, as you can see here, both of these, the metencephalons, and I kind of always think of it in terms of met has a T in it, and then the T has the cross. And if you notice, this area has these two big enlargements. There we go. One to the front and one to the back. And that's how I kind of remember that the metencephalon are these two enlargements. The first of these enlargements is this big region here known as the pons. Now, as I mentioned, we are in the brainstem. And one of the functions of the brainstem is to connect our spinal cord, or really all of our body, up into the cerebrum so we can process information and make sense of it. So if you notice, we had those cerebral peduncles. We have all these other enlarged areas where we're going to have massive numbers of fiber tracts. Information going in axons up and down towards our, actually, let me do this, towards the cerebrum and away from the cerebrum. Right? Now, if you think about it, if you're going towards the cerebrum, let's do this way. You are traveling up. And if you're going away from the cerebrum, you're traveling down. Of course, if we know anything about anatomy and physiology, we're not going to get away with terms like traveling up and traveling down. They're going to give us some kind of fancy vocabulary. And traveling up towards the cerebrum, we are going to call ascending information. And traveling down, we are going to call descending information. And so let's ask one more question. What type of information do you think is ascending? What type of information is primarily coming up to our cerebrum to be processed? Sensory, absolutely. So our ascending information is sensory, and therefore what type of information would be the descending information coming down? effect, or really motor, if you think about it. It would be therefore, it would be motor information. So our ascending information is going to be sensory. Our descending information is going to be motor. Excellent. Perfect. And so there here in our ponds, we have a massive number of fiber tracts going up and going down, right? There are going to be sensory ones that are going up, motor ones that are coming down. And so there's going to be big bundles of axons within this. The other thing we're going to see here is the pons also, another important place to send information to, as we'll see, is the cerebellum. We could practically spend half the semester talking about the cerebellum. A whole lot of stuff goes on there. But not surprisingly, there are a lot of axons that need to go to and from, actually I'll start with a highlighter, to and from the cerebellum. Well, remember up here, these big, huge knuckle-like bundles of axons going to the cerebrum were called the cerebral peduncles. Well, these three structures right here are what are known as the cerebellar peduncles. The largest of these is the middle cerebellar peduncle. So let's change our color. The biggest of these is the middle cerebellar peduncle. And this middle cerebellar peduncle is the, oops, sorry, I colored the wrong one. Wasn't even paying attention to what I was doing. Sorry, I apologize. I know this picture doesn't make it look like the largest one, but the largest one is this one here. This is the middle cerebellar peduncle, and it connects to the biggest axon bundle collection connecting the cerebellum to the pons. And I think I have a better picture that shows this. There we go. Notice here we see the three: superior, inferior, and the middle. And so here is that biggest one. Here is that middle cerebellar peduncle, which is basically the bundle tract going from the pons to the cerebellum. All right. Notice one more thing, I mentioned it, and again, we'll be coming back to it as well. But this diamond-shaped region in here is the fourth ventricle, the fourth fluid-filled space in our brainstem and brain. And as you can see, it is basically between the pons, which is anterior, and the cerebellum, that is posterior. If we change our view to this one here, notice here again, we see in between the pons, in between, there we go, in between the pons and the cerebellum is the fourth ventricle. And we see it both in the real brain and also the illustration of it as well. Now, our pons is not just fiber tracts, although there's a lot of them. There are also nuclei in here. Some of the most important ones, as you see here, are the pneumotaxics and the aponeustic. Any idea what pneumotastic and aponeustic refers to? They're big fancy bundles. You don't need to know the names of these centers. As you can see, these nuclei are not on your list. But I thought maybe pneumo in particular, because it sounds a lot like pneumonia, might give you a sense of what it is involved in. Where do you get pneumonia? Yeah, there you go, your lungs, breathing, absolutely. These are two major important nuclei that play an important role in determining your normal rate of breathing. All right. Let's talk a little bit about the cerebellum. Hopefully you have looked at the histology review that I posted. If not, shame on you. That's going to be a huge part of your lab exam. I guarantee you all of that is material that's going to be on there. And the first thing we talked about was the cerebellum. The cerebellum is somewhat unique in that it has raised ridges and it has grooves, just like the cerebrum. But unlike the cerebrum, the raised ridge is known as a folium, but the groove is still a sulcus. The other interesting difference between the cerebrum and the cerebellum is there are some similarities, as you notice. Let's do it this way. The gray matter is superficial, the white matter is deep. So this would be the white matter here coming out of this, or here that we see it here. In fact, the white matter, and I think you actually see it a little better here on the actual the gross anatomy, the dissection of it. The white matter forms this elaborate tree-like appearing structure with all these limbs and branches, and that's actually where it gets its name. It is actually referred to as the arbor vitae, the tree of life. Now, looking at the gross anatomy here, you don't see it, but what is distinct about the cortex and different about the cortex in the cerebellum versus the cerebrum is that in the cerebellum, there is not one, but two distinctly different layers of gray matter, two distinctly different layers of gray matter tissue. So one is more superficial, one is more deep, but both are obviously more superficial than the white matter. And the reason this is significant is because, as we talked about in the histology, here in the border between the two layers of white matter, the two layers of gray matter, is a very specific cell. Anybody looked at the histology yet? What cell is found here in between the two layers of gray matter in the cerebellum? I was going to do a histology daily quiz this morning to see who's looked at it. I guess apparently now I have to do it tomorrow, so be prepared for that. If you haven't looked at the histology yet, then you better today, because tomorrow you're going to have a daily quiz on it. I'll wait while someone tries to figure it out. You have another handout besides this handout, a histology handout. Now might be a good time to look at it and try to figure out what type of cell you would be looking for in the cerebellum. Purkinje, there you go, excellent. These cells are Purkinje cells. Perfect. Why, of course, are they called Purkinje cells? Good old Bob Purkinje was the first one who described it, identified it. Purkinje is what we affectionately like to refer to as an anatomy whore. Purkinje loved using the microscope. He was actually an amazing artist. And so what he would do is he would look at parts of the body under the microscope. He would draw these beautiful drawings of the things that he would see, and he would publish them. And of course, whenever he did that, he would plant his flag on them. So here in the cerebellum, you're going to learn about Purkinje cells. When we get to the heart in the cardiovascular system in 431, you'll learn about Purkinje fibers in the heart. I mean, he is all over the place. His fingerprints are all over the human anatomy, especially in the nervous system, but huge, huge anatomy whore. So again, this is the name of the cell. And obviously we have a location. That location is the cerebellum. But as we know, location, function, and structure go together. What do you think its function might be? The fact that it's found in the cerebellum, we have a location, and the cerebellum is part of the central nervous system. So considering it's part of the central nervous system, what do you think the function of this neuron might be? There you go. Well, integration, true, but remember, when you think of three functional types, they're sensory, motor, and interneuron. So the functional classification, you're right, its function is integration. But if we're thinking in terms of the functional classification, which I guess would have been better the way I say it, it's an interneuron. And if we know its functional classification, we know its location. Should we know its structural classification? What is the structural classifications of all interneurons? Multipolar, excellent. All right, this is kind of what I'm talking about on that review that's due on Monday. We need to be able to put together location, function, and structure. So when I give you one of those pieces of information, you know the others, right? And so here we have a specific cell, the Purkinje cell. We know its location, and based on its location, we were able to determine its functional classification and its structural classification. All right, if we have time, actually, no, I'm going to not show it to you histologically. You need to go and do that histology review. And again, this will be the last time I will warn you about it, but I guarantee tomorrow you will have a histology daily quiz. So make sure you have looked at that histology, you're familiar with the histology, because you are going to be responsible for that tomorrow on the daily quiz. All right, excellent. So we've talked about some of the basic anatomy. We have the white matter that's deep, forming the arbor vitae. We have the folia. We have the sulci. We have the gray matter. We have the white matter. We have all of that. One of the interesting things about the cerebellum, again, from an anatomy standpoint, I got to clear my drawing now so we can switch slides. Like the cerebrum, as we see here in the illustration, notice the cerebrum, as we know, has two hemispheres. Well, the cerebellum has two hemispheres as well. However, notice with our cerebrum, there is this huge invagination in between the hemispheres. What did we call that huge invagination between the two hemispheres again? Longitudinal fissure, excellent. Notice here, though, however, our two hemispheres of the cerebellum don't have an invagination. Instead, they have, I know when you look at the cerebellum here, it looks like a big pound of ground beef to me, or maybe ground turkey because it's a little light in color. But if you look closely, the illustrations make it easy. You can clearly see it here on the illustration. You can clearly see it here on the illustration. But even on the real cerebellum, you can see that there is this raised ridge. Now notice this raised ridge in between still has segments, so we can still see folia, we can still see sulci on it as we look at it. And because of that, it kind of looks like a segmented worm, which is actually where this raised structure gets its name. Right? The same way vermicelli gets the name of it as a noodle because it is a worm-shaped noodle. This raised segmented structure in between the two hemispheres of the cerebellum is what is known as the vermis. All right, that is all of the major anatomy we are responsible for the cerebellum. There is some microscopic anatomy you still need to look at. But the other thing is its function. So what the cerebellum does, and again, as I mentioned, the cerebellum does a lot of things, but one of the major functions, it has a major motor function in that it plays an important role in refining learned motor behaviors. This is not our autopilot, the same way we were talking about with the basal nuclei. What happens is as you learn a behavior, and again, walking is a great example of that, this refines that learned behavior, allows you to do that mechanism smoothly. Again, I mentioned one of the ways that you can see and understand how these areas work are when these areas are injured. There have been numerous instances of individuals who have been in a car accident. The police show up and arrive. They pull the person out of the car, and the person can't stand upright anymore. They're wobbling all over the place. The policeman asks them to walk a straight line, and they're stumbling as they do that. and they get arrested on suspicion of being driving while intoxicated. And they're denying it the whole time. They refuse to take the test. They get them to the police station. They draw the blood from them, and nothing. Zero alcohol in their blood. So then how do you explain the behavior? Well, what happens is a condition we call ataxia. Ataxia is a condition where you get a trauma to the cerebellum. This can often happen in a car accident, especially if you don't have the headrest set up properly. If your headrest is not set up properly, what happens when you get in a car wreck is you're often thrown back into that headrest. And if the headrest isn't set properly, instead of protecting your brain, it produces a trauma to your cerebellum. And that trauma to your cerebellum causes a condition known as ataxia. And that ataxia is that inability to maintain that refined motor behavior. So instead of being able to maintain posture, you're wobbling all over the place. Instead of being able to walk a straight line, you're stumbling all over the place. The good news is often when that occurs, it is a temporary condition. But in rare instances, if the trauma is severe enough, it can be permanent. Think how horrible that would be. All the wooziness, all the stumbling around of being drunk without actually getting to enjoy the alcohol, right? That would not be a fun condition or situation to be in. All right. So again, the cerebellum does a lot of other things, but one of its major roles is definitely helping to maintain and refine learned motor behaviors. Again, this doesn't help that 1-year-old as they're trying to learn how to walk. But once we learn how to walk, it's going to help them to maintain that stability. All right. Questions on that? All right. Excellent. One more cephalon to go. And that is the myencephalon. Again, here we see it a little bit closer. Again, they've given us this nice transparent appearance to it to show us that the medulla oblongata, the myencephalon, is again a major collection of fiber tracts and nuclei. One of the important things to remember is, or hopefully maybe you've noticed as we've been working our way through this, is as we go from the cerebellum down, as we descend the brainstem, the functions become much more basic. All right, one of the things that we would be doing, heck, we probably would be doing it today or tomorrow, would be a sheep brain dissection in the classroom. Our sheep brain and our brain is very, very different. However, most of the differences are in the cerebrum. As you look across species, sheep, cat, dog, monkeys, humans, the biggest differences in the brains are in the cerebrum. Their size, their shape, the number of folds in it, the orientations to them, all of those types of things are the parts that are dramatically different. But if you peel that cerebellum away, the brain stems, the parts that's left behind, like we have on the board here, is almost identical in all of these species. And so the anatomy of the sheep brain here is very similar to the anatomy of our sheep brain as well. Not our sheep brain, but our brain as well. Because they're serving basic life functions. Right up here is where we're doing our optical vision and our critical thinking and all of those things that the manual dexterity of our fingers that clearly a sheep can't do because sheep don't have fingers, right? But being able to maintain heart rate, being able to breathe, right? All of those types of basic structures, right? And that's here in the medulla oblongata, at the base of the brainstem is where most of our major biological functions occur. Things like there are cardiovascular centers to control our heart rate. There are respiratory centers here as well as in the pons for control our breathing. Many biological processes, coughing, vomiting, swallowing, hiccups, sneezing, all of these basic biological functions that you do and your dog does and your goldfish probably does, all occur down here in the medulla oblongata. And that's where we see with a bunch of these nuclei that are here. Now, I'm not going to make you know and understand the internal anatomy of this, but I do want a closer look at this picture. If you take a closer look at this picture, there's a couple key things I want you to notice about the medulla oblongata. The first is notice that many of our cranial nerves are associated with the medulla oblongata. So there's a color on one side and not on the other. We'll talk about this, like I said, I'm hoping today. This is cranial nerve 6 and 7 and 8. This is 9, 10, 11, and this one is 12. Now, I am going to put some small numbers next to these, and I'll explain why in just a minute. So again, this one here is 9. This one here is cranial nerve 10. This one here is cranial nerve 11. And this one here is cranial nerve 12. The reason I'm making a point of emphasizing this now, before we get a chance to talk about the cranial nerves, is because I want to talk about these two enlargements. If you look closely, change colors here, in between cranial nerve 12 and cranial nerves 9, 10, and 11 is an enlargement that sticks out. This big enlargement that sticks out here, and it is there as well. And notice here you get the name. This is the olive. The olive, as you can see, is the largest nucleus in the pons. And the olive is actually what is responsible for that sense we were talking about before, proprioception. Your olive is where your sense of balance and equilibrium and proprioception comes from. This is what allows you to maintain balance. This is what allows you to walk that line. This is what allows you to touch your nose, know where your body is in space, even when your eyes are closed. That is the olive that does that. Notice the second enlargement. Let's use blue. And it's located here. Oops, hold on. There we go. Between the two 12th cranial nerves. So this big enlargement right here and this big enlargement right here. So in between cranial nerves 12, one on one side, one on the other, is what is known as the pyramids. And these pyramids are where most of our somatic neurons cross. Right? One of the things we've been talking about since the very beginning of this class is we know that if I'm going to feel something with my right hand, I process that on the left side. If I'm going to move my right hand, I make that decision on the left side. As we know, most somatic neurons are contralateral. They control the opposite side of the body. And it is here in the pyramids where most of them cross. So it isn't so much a structure, a function, but here in this structure that is the pyramids, we know that there's an important thing going on. This is where the axons are crossing. All righty. Questions on that? All right, just that simply then, we are done with the brain anatomy. We still need to talk about the spinal cord, but I think we'll save that for the lecture. The other thing we do need to talk about are the protections. Now that we've talked about the brain, we can talk more about the protections, both the coverings, those meninges that we mentioned way back in the day, but also this will be our chance also to talk about the spaces that are filled with cerebral spinal fluid as well. However, before we dive into that, I think this is a good point to take our first break. So let's go ahead and take our first break here. Any questions before we take a break? All righty, let's go ahead and take a 15-minute break. It is 8:57, so we will restart at, that'll be 9:12. And we'll start the recording at that time. All righty. Let's go ahead and get started. We have talked now about our brain and brainstem anatomy. But to finish it off, what we need to do is talk about the protections, first of the brain, and then also a little bit later of the spinal cord, although we'll see they're very, very similar to each other. And also, those protections also include the fluid-filled spaces associated with it as well. All right. Now, as we look here at this pretty picture, there we go. Here we see collectively the protective coatings of the brain and spinal cord known as the meninges. Remember way back, really in the first week of this class, first couple days, we talked about we have all these organs inside the spaces of our body, and they are protected by layers of connective tissue. In our ventral body cavity, we talked about the serous membranes, both visceral and parietal, that provide protection. And in our ventral body cavities, our cranial and spinal cavities, we have what we called at the time the meninges, and we left it at that. Well, now we finally get a chance to come back and talk about what the meninges actually are. The meninges are actually a collection of 3 different protective layerings over the top of the brain and spinal cord. So meninges is actually the plural term, and the singular is the meninx, similarly to the way that we have phalanges, but the singular was a phalanx. Here we have 3 meninges, and each individual one is a meninx. I want to start first with the thickest of these protective layers. It is one that is known as the dura mater. Dura mater actually literally means tough mother. And it is made up of 2 connected layers of thick, dense, irregular connective tissue. In fact, it's a little bit more complicated than that, right? As you can see clearly by the amount of hair on top of the head, this is an example of my skull. But what we can see here is that we have the bone of the skull, right along the top here. And as we know, we have skin up here on top as well. And as we also know, there is a layer of dense, irregular connective tissue that surrounds bone. What would we call this? Oops, wrong direction. What would we call this dense, irregular connective tissue that surrounds and protects the bone? Periosteum. Excellent. Isn't it fun how as we learn things, we keep using them over and over and over again in this class? It's almost like the things we learned at the beginning were on purpose. Amazing that way, huh? Absolutely. So we have this periosteum that surrounds the skull bones, and the inner layer of that periosteum helps to form the dura mater. So one of the 2 layers of thick, dense, irregular connective tissue is actually the periosteum of the bone. And then there is a second layer that connects to it. If you think about it, this makes sense. This means that the packaging that is holding the brain in place is anchored to the bone of the skull, keeping it in place and hopefully limiting the amount of movement that is going to take place. But 2 important other things happen because of this as well. Notice at first, and let's switch to green. At first, these 2 layers are butted up against each other, continuous basically with each other, forming one very thick, tough mother. As I mentioned, this dura mater. If you're going to do brain surgery, you have to cut through these 2 layers to get to the brain to do the brain surgery. But these layers are so thick, so fibrous, so hard, you can actually sew them back together when they're done to help to form that seal around it. We have a pretty picture that I'll show you of this. And actually, I have one in the classroom that I usually get a chance to pass around, but obviously that's not something we can do here. But notice that inner second layer of the dura mater, while first is connected to the periosteum, eventually separates from the periosteum. And as it separates from the periosteum, it forms this thick dura matter that goes down and helps to stabilize the brain and hold the brain in place. Remember, I mentioned to you the brain tissue, nervous tissue, has the consistency of a jello jiggler. That's not very structurally sound. You don't necessarily want it constantly wobbling around like a jello mold inside of your head. So what happens is we have these extensions of the dura mater, and there are three you're going to be responsible for. Extensions of the inner layer of dura mater that form protective structures that help to provide additional support for the brain in the cranial cavity. And this particular one, the one that goes down the longitudinal fissure, is known as the falx cerebri, because it sits on the fault line, the falx between the two hemispheres of the cerebellum. So it forms that falx cerebri. So one of the important functions of the separation of the two layers is to form these additional folds. And we've got a great picture where all the pesky nervous tissue is removed and we can just see the dura mater and how that functions to form individual spaces that stabilize the brain inside of the skull. But notice, and even though it's blue in here, I'm going to use red.
Professor?
Yes.
Can you move just a little bit higher the writing where you wrote it?
Sure. How's that?
Okay, yeah, perfect, thanks.
Okay. The other thing that I want to point out to you is that notice when the two separate up here, it forms a big fluid-filled space. This big fluid-filled space actually has a very special name, and we call this very special structure a sinus. Did I spell it right? A sinus is basically a very large, loose vein. Why might it be important to have very large, loose veins?
To bring nutrients to the brain and stuff like that? Or...
True, but is that really what veins do? Do veins bring blood to the brain, to the skull?
No, away from.
Away from. Veins bring things away from, right? Let's ask the question in a different way. If, again, when class gets done today, you are super excited to, I don't know, go shopping or whatever it is, you get to leave the house for some exciting reason. And so you go running out, and in your haste, you slam the car door on your hand. As a result of that injury, one of the things that happens is it will swell, right? That edema, as excess blood and fluid is released into that tissue, causing that swelling, right? Is swelling to your hand a huge deal? Right? Maybe it makes it a little harder to take your ring off when you get to the bar at the end of the day, right? Maybe your joints feel a little bit tight. I mean, it's not going to be comfortable, but is a little swelling in your hand, again, I'm not talking about the damage. Obviously, the damage is an issue. But if we have edema in our hand, is that a major issue?
No.
No, typically not. Absolutely. But that's because this tissue can expand. But what about here in your cranial cavity? What if we start accumulating excess fluid in our brain or surrounding our brain? Could that be a problem?
Yes.
Yeah, because our skull doesn't expand the same way that our skin and our muscles do here. You can get a major increase in pressure and cause major serious damage to that nervous tissue. So one of the things that we need to be very efficient at is draining fluid from our cranial cavity. And by having these big, large, loose veins, these sinuses, it allows us to accumulate all of that fluid, all of that blood, all that cerebral spinal fluid, and get it out of the skull as efficiently as possible to reduce the chance of that intracranial pressure of that swelling, that edema that could occur in there. All right? So again, two huge advantages for the separation of that. It helps to provide much structural integrity, like with the falx cerebri, but it also forms these spaces for these big loose sinuses to help us to drain the cranial cavity as efficiently as possible. All right, that is our dura matter. Let's talk about our second menax, and that is the arachnoid matter. Arachnoid matter, of course, gets its name because of its web-like appearance. Oops. It, again, I appreciate that this tissue, the artist here, has drawn, color I haven't used yet, this kind of continuous layer here on top, and then all the fibers coming out from it. But it really, the arachnoid matter really doesn't have that much structure to it. It really is kind of like the fibrous web of a spider, which is where it gets its name, arachnoid matter. And its primary job is to form that space, and that space, that cavity, is the subarachnoid space. And this subarachnoid space is going to be filled with cerebrospinal fluid. All right. So this space, again, the outer surface of our brain is bathed in cerebrospinal fluid to provide some protection, right? One of the ways that we protect our brain from rattling around is surrounding it with fluid, right? If you just jumped into your bathtub right now and a huge earthquake went on, right, you'd get rattled around inside of that bathtub, bouncing off the walls. However, if that bathtub was filled with water, right, yeah, when that earthquake hit, water would go splashing out all over the place, but you would be cushioned inside of that, and you wouldn't be thrown against the walls as roughly. And that's basically what it does. By bathing the outer surface, it adds some buoyancy, taking some of the pressure off of the nervous tissue, and then it also protects it from movement as well. All right. Now, there's one more point I want to make on this, and I'm running out of spaces to write this, but I'll try to sneak it in as best as I can. Right here, no, let's not use that color. Right here, between the dura mater and the arachnoid mater, is a potential space. Someone remind me again what a potential space is? Anyone remember? That's all right. I wasn't paying attention either. This potential space is called the subdural space because it's underneath the dura and between the dura and the arachnoid mater. And maybe you can answer this question for me. What is in the subdural space? Someone tell me what is in the subdural space. No, not spongy bone. Well, it's a potential space. No, not fat. Not cerebrospinal fluid. Those are all great guesses, but none of them are correct because this is a potential space.
So there's nothing?
Nothing. There is nothing in this potential space. Again, if you remember the example I used when we talked about a potential space, it's if I take my hands and mash them up against each other, do they fuse together to become just one structure? No, of course not. I still have two hands. They're just next to each other, mashed up against each other. So technically there is the potential for a space between them, but as long as they're mashed up against each other, it's just a potential space. And what's in between my hands? Nothing. And that's what's in the subdural space as well. The dura sits right on top of the arachnoid matter, and there is nothing in between. At least there's nothing in between in a healthy brain. What happens if you have forgotten to take out the garbage for the umpteenth time, and your significant other has finally had enough, and so takes that hammer and hits you in the head with it? As a result of that, there may be some bleeding that occurs. And one of the things that sometimes occurs is that bleeding may accumulate in that space between the dura and the arachnoid matter, and it would fill with blood. Again, it's not there in a normal healthy brain, but now we have that damage that has occurred, and we have an accumulation of blood in that area. And what would we call that? I know you guys know this.
Are you talking about like an inflammation or?
Nope, I'm talking about accumulation of blood between the dura mater and the arachnoid matter. It is edema, but this is an accumulation of blood, so there you go. It is a subdermal hematoma. Exactly. Right? Hopefully none of you have one of those right now. Hopefully there is nothing between your dura mater and your arachnoid matter, because it is just a potential space. But it is a space, even if it's potential. So if there's an injury or if there's damage or something like that, things can accumulate in there. But in a normal, healthy brain, the two are mashed up against each other and there's nothing in between. All right. Excellent. Now, notice there is a third layer to our meninges, and our third meninx is what is known as the pia matter. The pia matter, and actually I'm going to cheat here. Hold on, I want this space, so I've got to move my windows. And move that there. So that's for there. And then over here I can then write. Our pia matter is basically our shrink wrap. It is directly on the surface of the brain. Notice not only is it directly on the surface of the brain, but notice also some of its fibers actually go down into the sulci, holding them together. Again, one of the things that you would have had the opportunity to do if we had done a brain dissection, if we were on campus, is you can actually peel the pia mater off. And when you peel the pia mater off, you're actually able to open up the sulci and separate the gyri without actually damaging the tissue. It also helps to stabilize and hold blood vessels in place. So this is the shrink wrap stabilizing the nervous tissue, stabilizing the gyri, and holding blood vessels in place. And so these three layers together, the dura mater, the arachnoid matter, and the pia mater, form those three layers, those meninges that surround and protect the brain, as well as forming that subarachnoid space where we are going to then maintain our fluid. All right, our cerebrospinal fluid. Questions on that? All right, the good news is when we get to the spinal cord is going to have those same three layers of protection on them, but there are also going to be some subtle differences as well. And so we'll see that because of the differences of the function between our vertebral column and our skull. All right, with this in our pocket, any questions on the first part of the protection, and that is our meninges? All right, let's talk then about our second layer of protection. Actually, before that, we got the pretty picture from your textbook. Notice here we have a intact skull, so we actually have some hair. We have some of the bone, the scalp. And again, notice over here, we have this thick, fibrous, opaque looking layer that is the dura mater. That, as you can see, has to be cut to be removed. And notice there really isn't a lot of arachnoid matter here. That arachnoid matter is more fiber. So when you peel off the pia mater, the arachnoid matter pretty much comes with it. You see a little bit of the arachnoid matter fibers down here by the spinal cord in this region, but not a lot. But notice what you do see is you see those big, huge dural sinuses that we talked about. Those big, huge, loose veins that are collecting that blood and pulling it out. Notice when we look at the surface of the cerebrum, it is very shiny in appearance. And the reason it is shiny in appearance, has that reflection to it, is because it has that pia mater on top of it. If we peeled off the pia mater, and here, actually, let's go back. Oh, actually, I have to do this first. All right, so hold on. Oops, cancel that. I want to stop sharing this for a second. Cheat and go back to, because I closed it already. Here we go. Remember again, when we first looked at the brain, here, this shiny surface stabilizing the blood vessels in place, making it shiny, holding the gyri together so that the sulci stay closed. Here, we have a brain that still has its pia mater on it. All right. However, if you remember when we looked ahead a couple slides, here, we can see this brain has had its pia mater removed. Most of the superficial blood vessels are gone. Notice there are still some deep blood vessels that we see in here. But notice now that all of the sulci are open. They're more open. Notice we didn't damage the tissue in removing that pia mater. We just carefully removed that shrink wrap, and the whole brain kind of opens up as a result of that. So there we can kind of see the difference between the two of that. All right. So now I can close that again. And we can come back here. Come on, there we go. And one more picture just to beat the dead horse. Here, notice we can see again the layers of our meninges, starting first with our dura mater. And again, notice we have 1, 2 layers to that dura mater, and then that sinus in between. Here we have the arachnoid mater with all of its fibers. And notice also here in between, we have that subdural space, that potential space between them that does not contain anything. That is different from the subarachnoid space, which contains cerebral spinal fluid. So again, the subdural space contains nothing. It is just a potential space. Whereas our subarachnoid space contains cerebrospinal fluid. Excellent. And then last, but definitely not least, we have that shrink wrap pia mater on the surface of the brain. And notice also, as we mentioned, this superficial layer of the dura is basically the periosteum of the bone. Now, as I also mentioned, we did that, we'll come back to that. That deep layer of the, that deep layer of the dura comes off and separates from the superficial layer to form 3 different folds that help to stabilize and hold the brain in place. So here, that pesky brain and spinal cord have been removed, So we just see the protective coverings. Notice in these protective coverings, we can see the large sinuses. Here's a sinus, here's a sinus, here's a sinus, here's a sinus, here's some spaces for sinuses, more sinuses, all of these together. And don't worry, I know you feel bad because you don't have to learn them right now, but when we get to the cardiovascular system in 431, you will have to learn the name of all of these individual sinuses. But again, the point of these is all to have these big, huge, loose, open blood vessels so that we can drain blood out of the cranial cavity as easily as possible. So I know you feel like you're missing out a little bit, but don't worry, we'll get there eventually. But what we do need to know for this exam is the name of these three folds. Notice, as I mentioned, the falx cerebri is the largest of these extensions, and it sits in the longitudinal fissure between the hemispheres. Of the cerebrum. Excellent. Therefore, not surprisingly, the falx cerebelli that we see down here, this one right here, is going to sit in between the hemispheres. Of the cerebellum. Now, remember, the longitudinal fissure is a big, huge, deep invagination between the two hemispheres. If you remember, what sits between the hemispheres, the two hemispheres of the cerebellum? Right, not an indentation, but a raised ridge known as the vermis. So basically, this sits on top of the vermis. And as you can see, it's not nearly as extensive. It's much narrower because it, rather than going deep into a sulcus or fissure, it has to sit on top of a structure. So it's still going to help to stabilize the hemispheres, but it's much, much smaller because of that pesky vermis that is in the way. Lastly, as we see here, there is an extension of the dura mater that basically sits like a tent over the top of the cerebellum, stabilizing and separating the cerebellum from the cerebrum. And that tent sitting on the top of it, not surprisingly, is known as the tentorium cerebelli. So this tentorium cerebelli basically sits between the cerebrum and the cerebellum, helping to stabilize both of them. So these are three extensions of the dura mater that have names, which means you need to know them and know where they're located and kind of what they do. All righty. Questions on that? All right, excellent. Come on. Oh, I did that. Yeah. All right. Last part of the protection for the brain that we need to talk about is we need to talk about the fluid-filled spaces. We have already identified one of those fluid-filled spaces, and that, of course, is the subarachnoid space. So all of these are spaces that contain, well, let's actually emphasize that. These are all spaces. So on the exam, if I point to any of these things, I will say identify this space, because these are spaces found inside of the nervous tissue. So that is, and of course, they all contain cerebrospinal fluid. Now, excellent. If you've looked at the list, the way I have written it out is to talk about the flow of the cerebrospinal fluid. One of the important things to remember is there isn't just one distinct flow to the cerebrospinal fluid. As we can see from this illustration, there are 4 different ventricles. And each of these ventricles have the structure capable of making cerebrospinal fluid. So if you think about it, there are 4 different locations where cerebrospinal fluid can be formed. If there's 4 different locations where it can be formed, then can there just be one path that every single drop of cerebrospinal fluid takes? No, of course not. But as we can see from these structures, there are limited locations and only limited spaces and paths that they can take. So we will talk about, we will start as a drop of cerebrospinal fluid out here in one of the lateral ventricles, for instance, and follow the path that it can take. But it is important to know that all of these spaces have structures that will allow them to produce cerebrospinal fluid. Now, if we're going to talk about the formation of cerebrospinal fluid, the first thing we need to do, and I'm going to go ahead and cheat and steal some of the board here so that I can write on it. So let's make a white box right here so that I can do a little writing. Every single one of these, here, and actually I want to erase that first. Every single one of our 4 ventricles. So we have 4 ventricles. Let's start at the beginning. And as I mentioned, all 4 ventricles have the ability to produce cerebrospinal fluid because all 4 ventricles contain a special structure called choroid plexus. And that choroid plexus is what is used to make cerebrospinal fluid. Basically, cerebrospinal fluid is filtered blood plasma. One of the things we've been talking about since the very beginning of this class, as we talked about for maintaining homeostasis, is what is in our blood is in our interstitial fluid. The two are constantly mixing back and forth. However, as we've talked about, in our nervous system, our nervous tissue, it doesn't regenerate. So we have to be protective of it. So for instance, in the blood supply, we know those astrocytes form a blood-brain barrier, limiting what can get out of the blood plasma into the nervous tissue to help to protect the nervous tissue. If we're going to bother having those astrocytes forming that blood-brain barrier, then we can't just let any old thing come out of our blood vessel to form the cerebrospinal fluid. So we need a way to protect it as well. Obviously, that choroid plexus is going to be made of capillaries. But if we wanted to limit what was able to come out of it, it would be really convenient if we had one of those neuroglial cells that was almost like an epithelial tissue that could line the surface of those blood capillaries, helping to regulate the flow and monitor the flow of cerebrospinal fluid coming out of those capillaries. Did we have a particular neuroglial cell that might meet and serve that purpose? Well, astrocytes form the blood-brain barrier. But the ependymal cells, whoops, were the ones that were the epithelial tissue that wrapped around the outer surface and, as we've said, helped to form and monitor our cerebrospinal fluid. So basically, choroid plexus is this kind of chewing gum looking stuff that is basically big fancy capillaries wrapped up in ependymal cells where the cerebrospinal fluid can come out of it. And all 4 of our ventricles have them. In our lateral ventricles, and those are these 2 horns right here, it lines the inferior surface of our lateral ventricle there. In our 3rd ventricle, which is this one here, it lines the superior surface of that right along the top wall. And in our 4th ventricle, It basically sits here behind the pons, in between the pons and the cerebellum. So all 4 of our ventricles are going to have a choroid plexus inside of them. So cerebral spinal fluid could be produced in any of these 4 locations. All right, let's take a different look at this. This is obviously a sagittal view. Here we look at it from a frontal view. Here we get a better sense of those 2 lateral ventricles. As we mentioned, there is a thick layer of connective tissue that separates the 2 lateral ventricles from each other, right underneath the corpus callosum. Anybody remember what we called this particular structure that separates the 2 hemispheres from each other? Test is 1 week from today. No, again, I know I've drawn the longitudinal fissure, but where this would really be located would be here in between, but I didn't want to confuse it with the 3rd ventricle. So here, let's just clear the drawing. There is an extension of connective tissue that separates the 2 lateral ventricles from each other. It happened to be between the corpus callosum and the fornix, right in that space in between. And what did we call that structure that was on your list that you were responsible for that we did in the last class? I think tomorrow's daily quiz is actually going to be 10 questions, 10 points. And not only am I going to put histology, but I'm going to put some brain anatomy on there too. Maybe it'll be a good precursor to the test that's going to be going on a week from today. Encourage you guys to look at this stuff a little bit more. No, close. There it is, the septum pellucidum. Excellent. That septum pellucidum separated the 2 from each other. Let's try a different question. Notice here, notice the 3rd ventricle kind of has this kind of bird's beak, bird's head, duck head appearance to it. Because remember, it was that space in between the 2 thalami and the 2 hypothalami. So it was in that space in between. But notice there is a void. Oops, no, not what I wanted. That. There is a void right here in the center of our 3rd ventricle, indicating that there isn't a space there. Because, as it turns out, there was a structure in the center of the 3rd ventricle, almost like an island in the center of our 3rd ventricle, which is a bundle of axons that connects the 2 thalami together. So that void in the 3rd ventricle is because of that axon bundle that connects the 2 thalami. And what was that called again? Intermediate mass. Excellent. Perfect. All right. Notice if we switch to this view, although you see it a little bit here as well, but I think it looks a little better here. Notice that our lateral ventricles connect to our 3rd ventricle by a small fluid-filled space that connects them together. So again, if we go back to the previous picture, we see it right here. From this picture, we see it right here. And this small space that connects the lateral ventricles to the 3rd ventricle is what is known as the interventricular foramen. Here, of course, is our 4th ventricle. Here is our 3rd ventricle. There is a thin, narrow passage that goes through the midbrain, connecting the 3rd ventricle to the 4th ventricle. And that is that aqueduct of the midbrain. Or remember what we also called the cerebral aqueduct. Both of these are appropriate terms. And then you're into the 4th ventricle. So notice if you are a drop of cerebrospinal fluid, and as a drop of cerebrospinal fluid, you are produced right here in the lateral ventricle. From this lateral ventricle, there is only one place you can go. When you leave this, there's only one door out. That one door out is the interventricular foramen, and only one location you can be in at that point, the third ventricle. From the third ventricle, there's only one way out, down the cerebral aqueduct. Technically, this is a door here, but this is a one-way door. Fluid just coming in. Fluid's being made and built up there, so it's coming out. So we don't want to go in there. There's only one way out. Cerebral aqueduct to the fourth ventricle. So notice as we go through the flow of the cerebral spinal fluid, if you start in the lateral ventricle, there's only one place to go. If you start in the third ventricle or come into the third ventricle, there's only one place to go. Notice it's not here till you get to the fourth ventricle. That, conveniently enough, based on its name, there are 4 ways out. Notice over here is a hole to the side. This hole to the side is what is known as the lateral aperture, because it's an opening to the side. And there's one on each side. Notice there's also this tiny hole right there. That is actually an opening that comes out posteriorly on the midline. And if these are known as the lateral apertures, guess what the one on the midline is known as? Medial aperture. Excellent. So you can come out that median aperture or medial aperture. Or the fourth place it can go is down the center of the spinal cord. Down the center of the spinal cord is a structure known as the central canal. And so this not only is there cerebral spinal fluid inside the brain, there's cerebral spinal fluid inside of the spinal cord as well. I think this picture does a really nice job of showing these 4 exits to the fourth ventricle. If we go back to the previous one, I got to clear my picture for that. Notice it emphasizes it sort of in a similar fashion. We have obviously the fourth ventricle that can go down this way. We have, it shows one of the lateral apertures where it can come out this way. And then this represents the third ventricle coming out that way. But I think the picture that shows these the best, aside from that frontal view, is this one here. So notice on this picture here, what's nice about this is not only does this show the exits of the fourth ventricle, but it shows where things go. All right, so notice we come into the fourth ventricle through the cerebral aqueduct, and there are 4 ways out. One way out, right on the midline, is the median aperture. Notice when you come out of the median aperture, you are basically in the subarachnoid space below the cerebellum. Let's make this a little smaller so it all fits on the screen. Excellent. So if you come out the median aperture, you're in the subarachnoid space below the cerebellum. Notice they've given us an indication of where it is. It is superior and obviously lateral, but because it's going out to the side, we really can't see that it's going out, but it is going out to the side. And notice when it goes out to the side, that cerebral spinal fluid enters the subarachnoid space above the cerebellum. And remember, there's 2 of those. And the fourth place where cerebral spinal fluid can go is down the central canal of the spinal cord. However, notice the spinal cord doesn't go down forever. Eventually it ends. And when it ends, our cerebral spinal fluid is now in the subarachnoid space around the spinal cord. So notice while technically you do have choices from that fourth ventricle of where you go, notice that all cerebrospinal fluid eventually ends up in the subarachnoid space. So notice also, while we produce cerebrospinal fluid constantly, we don't produce any of it in the subarachnoid space. It's just produced in the ventricles, but it does fill that subarachnoid space, right? And again, fill is a relative term. During the course of the day, you produce somewhere around 500 milliliters in one day of cerebrospinal fluid. And at any one time, you have around 150 milliliters in that space. So it's not a massive amount. So we're not talking about oceans of cerebrospinal fluid. But it is filling the spaces. And when we think of it, again, one of the big keys is to provide some cushion. But that doesn't necessarily explain why you would produce it on the inside. Producing it on the inside isn't providing that cushioning, because since it is filtered blood plasma, it contains nutrients and oxygen that the brain needs. It can remove wastes from the nervous tissue, and it contains chemical signals that the brain can monitor, like how much calcium you have in your blood, like what the pH of your blood is. Hormones, things along those lines can all be in the cerebrospinal fluid and therefore can influence the function of our brain. All right, so far so good? All right, excellent. We have now produced cerebrospinal fluid. We know the function of our cerebrospinal fluid. We know how it's going to flow through the brain, through the nervous tissue, out into the subarachnoid space. But if we're constantly making this, the other thing we need to do is we need to drain it. If it comes out of the blood vessels, it needs to go back into, needs to re-enter the blood vessels. And in particular, it needs to re-enter into those big blood vessels that are going to help to drain our cranial cavity. So it needs to enter into our dural sinuses. And it does that with a special structure. We see it in a tiny picture here. Notice, highlight it, and let's use green. There is this finger-like extension of the dura mater, the arachnoid matter. These finger-like extensions of the arachnoid matter. And remind me again what that term way back we learned in the skin for a finger-like extension was again. Be able to make our fingerprints. What do we call that? What's a term we use for a finger-like extension? A villi or a papillae, exactly. And that's what these things are called. They're called the arachnoid villi. They're called the arachnoid papillae. And they are also referred to as the arachnoid granulations. 3 different names, all the same things. You may use any of those. But what they basically are extensions of the arachnoid matter into those dural sinuses so that the cerebrospinal fluid can drain. And we quickly breezed over the picture of this before, so let's take another look at it again here. I think I have it as the next slide. Nope, I lied. Oh, I don't like that. Let's fix that. There we go. Notice this was the picture we looked at before. We have the two layers of the dura mater with the sinus in between. We have our arachnoid matter, the subarachnoid space, the pia mater, pardon me, the subdural space, the subarachnoid space. And here we see this extension of the arachnoid matter. The arachnoid, again, granulation, villi, papillae, all of those are acceptable terms for it, where our cerebral spinal fluid is able to enter back into the blood and continue the cycle. Now, as I mentioned, your book's got some great pictures of these things. Notice here we see in this nice weird kind of transverse, partial transverse section, we can see the C-shaped lateral ventricles. And notice here, we can better see that septum pellucid that we talked about that separates the two lateral ventricles from each other. So we see that really nicely here. Oh, and notice also the other thing that we see here is that, right here we can see that choroid plexus, those ependymal cells on top of capillaries lining the inferior surface of our lateral ventricle, producing that cerebral spinal fluid. Here we see another nice illustration showing the flow of the cerebral spinal fluid. Again, notice it can be made here in the lateral ventricle. It can be made here in the third ventricle. It can be made here in the fourth ventricle. Where no matter where it's made, eventually it ends up in the subarachnoid space, and then it ends up back into the blood supply. And lastly, that picture that I showed you already, notice again, as we talked about, here is the view of the metencephalon with that pesky cerebellum removed. And notice now, not only do we see here is our triangle-shaped fourth ventricle, but notice here to the side are where the lateral ventricles, lateral apertures are located. Here, right here, is where the median aperture is located. The fluid would also continue down into the central canal of the spinal cord. And notice right here, this red chewed up piece of chewing gum looking structure here in the center of our fourth ventricle is our choroid plexus. Oh, and notice we also see the choroid plexus that lines the bottom of the lateral ventricle as well. All right. So those are all of the structures involved in the production and the spaces associated with the movement of the cerebral spinal fluid. Any questions on that? All right, excellent. With that, we are done with anatomy of the brain, and we can move down to the anatomy of the spinal cord. Remember, we're still in the central nervous system. The spinal cord, along with the brain, is part of the central nervous system. And remember, as part of the central nervous system, its function is integration, processing the information, and making decisions. And our spinal cord absolutely positively does that for us. Now, does that mean that it is your spinal cord that decided who you were going to marry, or what job you wanted to be, what you grow up, or even what you decided to have for breakfast this morning? Are those the types of decisions that our spinal cord is making? No, of course not. It is making much more basic decisions, right? Simple decisions. In fact, we kind of have a fancy term for the types of decisions that our spinal cord makes, right? Because if you think about it, while it didn't help me decide what I had for breakfast, because I had eggs, but when I put my hand on the hot stove, right, I pulled that hand away from that damaging stimuli before I was even aware of the fact that I should be cursing. And what do we call that type of decision-making process? Reflex, absolutely. So many of our reflexes take place in our spinal cord, right? Where it is making basic decisions, right? This is painful, I should move away from it. And it does that, like I said, before I'm even consciously aware that it is occurring, absolutely. So it does absolutely make decisions. It is part of the central nervous system. It is integration of information, right? So it is processing, it is bean counting. That's what we know neurons do. They receive positive or excitatory postsynaptic potentials, inhibitory postsynaptic potentials, and depending on how many of both and where they happen in time, it decides whether it's going to fire an action potential or not. But obviously, one of the other major functions is to be that highway or byway of information. And again, I already explained this, but let's go ahead and do a quick drawing of it just to make sure we understand. Mostly just because I like drawing brains. There's my brain. There is my spinal cord. Excellent. And as we know, some information comes into the spinal cord, some information comes, oh, let's actually be clever about this. Some information goes out of the spinal cord. Actually, let's be really clever. Hold on. Undo, undo, undo. Actually, let's just do. Some information comes into the spinal cord. And some information comes out of the spinal cord. Let's pretend those were straight lines. What type of information comes into the spinal cord, into the central nervous system? What type of information comes into the central nervous system? Sensory, excellent. Sensory information. And what direction did we say that was? Going in towards the central nervous system was afferent. Excellent. Whereas information coming out of the central nervous system was what type of information? Motor. And what direction did we say that was? Efferent. Excellent. Excellent. So into the central nervous system, out of the central nervous system, afferent and efferent. We know decisions are made in the spinal cord, but as we also talked about, we know that the spinal cord is responsible for sending information down from the cerebrum, and it is also responsible for carrying information up to the cerebrum. And as we mentioned earlier, it is, of course, sensory information that is carried up to the cerebrum in the spinal cord. And what type of information did we say that was? When it goes up, we said it was, well, it is sensory, absolutely. It is sensory information, but that directionality, the direction of going up the spinal cord to the cerebrum would be, now afferent remembers into the central nervous system, so going up would be ascending, right? Ascending information going up to the cerebrum. And then as we mentioned, of course, then it is motor, information that comes down from the cerebrum. Notice we can't use ascending, we can't use afferent and efferent because those are into the central nervous system and out of the central nervous system. Spinal cord is the central nervous system, so we still can't use that. So instead it's ascending for sensory, and motor coming down from the cerebrum is descending. Excellent. So sensory information is afferent and then ascending. We process that information, make voluntary decisions, and then that information descends and is efferent to our effectors. All right. Back to there. So it is definitely that highway and byway. And again, I always think of it in those terms. It's this big, huge, massive structure, like one of those big, huge transatlantic cables carrying information from us here to England. But honestly, it's really not, if you're actually to hold a spinal cord in your hand, it really isn't nearly as impressive as one would think. I normally have my yardstick in the class. I don't actually have a yardstick here. Do I have a ruler? I don't even have a ruler. I have to have that for next semester. 16 to 18 inches. That's it. In fact, your spinal cord actually stops growing at age 4. How many of you reached your final height at 4 years of age? Anyone? Again, I can't look around the classroom and tell for certain, but did anyone reach their final height at 4 years of age? Didn't stop growing at that point? No. So notice what happens is you continue to get taller, but your spinal cord doesn't get longer. So in a mature adult, if you notice from the illustration, your spinal cord doesn't actually run the entire length of your vertebral column. Instead, it stops right about the L1 through I'd say 3, L1, L2. Actually, let's say 2. I like 2 better. L1, L2 region of the vertebral column. It actually stops. And all of these are just loose axons traveling down below that. So again, it typically ends at the L1, L2 region of the spinal column. It is a narrow structure, but there are 2 enlargements. One enlargement is a cervical enlargement. One enlargement is a lumbar enlargement. Why might there be 2 regions of your spinal cord that are bigger, thicker, larger than the other parts? Well, what's it made up of? Like all nervous tissue, it's made up of gray matter and white matter in our central nervous system. So cell bodies and dendrites and axons. So in an area that was larger, you would expect maybe it has more cell bodies, more axons. Why might you have 2 regions that has more cell bodies and more axons in it coming into and out of it? All right, why would there be more nerves coming into and out of the cervical enlargement, for instance? What do you think that enlargement is associated with? Yeah, the arms. You have far more nerves going into and out of your arms than you do the trunk of your body. And that means the lumbar enlargement is associated with your legs. There you go, exactly. Excellent. So we have those 2 enlargements associated with those. Let's take a closer look at the inferior end because there's some interesting things that happen down here.
Professor?
Yes.
Sorry to interrupt. Quick question. You were mentioning like the spinal cord is like it goes up to 18 inches. What about people that are like 6, 7 feet tall? What happens to those people? Is that an exception? They have it longer or?
That is a great question. Typically in those individuals, it is going to be a little bit longer. And again, part of that is that if you think about it, if a person ends up being 7 feet tall, then even as 4-year-olds, they're typically much larger than a typical 4-year-old would be. So their bodies have already been growing longer to begin with. But the spinal cord will typically be in a similar location. It may be slightly shorter in those individuals, depending on when they had their massive growth spurt. There's another implication to those later growth spurts that we'll talk about in just a second as well. But for most people, it is similar in the location. If someone's really tall, it might be a little shorter, but not much, because typically their increased growth is something that has been continuous through their life.
Okay.
Yep, great question. And you never have to apologize for interrupting. Like I said, it's not as easy here because you can't just raise your hand and let me know. I know technically you can raise your hand and I can see it, but I get talking and I don't always see the hand raising when people click it. So please, when you guys have a question, the whole point of me doing this is so that you can understand this information. I want to let you. Yeah,
I do have one more question.
yeah, go ahead.
I know I heard like stories and rumors and stuff like that, and I know like back in Europe, they were talking about that the spinal cord has. Are we talking about the same fluid where it's like very like if you take the spinal cord out of somebody, inject into yourself or something like that, like it gives you longer health or life or something like that? Do you know anything about what I'm trying to say?
Um, yes, and again, so here's what I would say. There has long been an infatuation with staying young, staying youthful, right? Extending our lives. Since medieval times. In medieval times, one of the things that certain monks in certain um, regions would do is that the males would drain some of the blood from young females because they believed that if they were to inject their blood, the young blood from the young females that had that youthful essence to it, it would help to extend their lives as well if they were doing it that way. And it's the same kind of belief this way. No, the thought is that uh, potentially someone who was younger and would have more immature cells in their cerebral spinal fluid, which would help to maintain an older person's nervous tissue or something along those lines. But the cerebral spinal fluid, as I mentioned, is just filtered blood. So it doesn't have any type of special cells or any other special capacities to it. So you don't gain any benefit from injecting cerebral spinal fluid than you would from injecting, somebody's blood into your body. If you take someone else's blood and inject it into your body, you don't get their memories, you don't get their, superpowers, you don't get their health, their youth, or any of those types of things. And it's the same thing with any of these humors of the body.
Okay.
Yep. All right. So, as I mentioned, the spinal cord doesn't just terminate. If you notice, what ends up happening is it tapers at the end, becoming more and more narrow, forming a cone-like structure. And this cone-like structure is known as the conus medullaris. And like I mentioned, it terminates at the L1, L2 region of the spinal cord. However, notice that doesn't mean that from the L2 region down, there is nothing in the vertebral column, because what we have is we have all the axons that are coming into and coming out of the spinal cord. And those axons continue to project downward till they finally get to the destination where they're going to leave the vertebral column, either the intervertebral foramen or they'll come out the sacral foramina. Right? Those are terms that we're familiar with, right? If you come out between the vertebrae, that's the intervertebral foramen, or you can come out the hole in the sacrum, that's sacral foramina where they come out. These bundles of axons extend down in a large, coarse, hair-like structure that is known as the cauda equina, because it looks like a horse's tail. It's called cauda equina because it looks like a horse's tail. You may not use the term cauda equina on the exam. You must use the correct term, cauda equina. So don't say horse's tail, say cauda equina, but that's what cauda equina means. It looks like a horse's tail. And that's just those bundles of axons extending down to where they need to leave the vertebral column. However, as I have hinted at, the same protections that are on the brain are on the spinal cord as well, including that shrink-wrapped pia mater. That shrink-wrapped pia mater also covers and protects the spinal cord as well. However, one of the interesting things that happens is the collagen fibers from it actually extend down and connect to the coccyx, forming a stabilizing structure known as the filum terminale. Now, remember, as we were talking about, your vertebral column continues to grow as you get taller. So as you get taller, it gets longer. But as we mentioned, your spinal cord stops growing at 4 years of age. So after 4 years of age, as your body continues to get longer, your spinal cord moves further and further up the vertebral column. And this extension of the dura mater, or more, I'm sorry, extension of the pia mater, helps to stabilize it and hold it in place. The problem is if you have a rapid growth later in life, I use the example of my brother-in-law who grew 12 inches his senior year in high school. One of the concerns, and thankfully this did not happen to him, but one of the concerns is, as we know, these collagen fibers aren't very flexible. They don't have a lot of give, and it's not easy to grow more of them. So what can happen is as your body gets longer, this filum terminale doesn't get longer fast enough. And so what happens is it starts pulling your vertebral column, pardon me, starts pulling your spinal cord down inside of your vertebral column. And as your spinal cord gets pulled down, one of the things that happens is you can start to kink some of the nerves as they're leaving the spinal cord. It can cause intense pain. It can cause loss of sensation, weakness in the appendages, things along those lines. It is an incredibly uncomfortable condition when it occurs, but luckily there's a really easy fix for it. What do? How do they fix this debilitating condition? Cut it, exactly. They basically go in and cut the filum terminale. Cut the filum terminale, the spinal cord goes to its correct location, and symptoms go away almost instantaneously. Now, of course, by cutting that filum terminale, you are no longer stabilizing the spinal cord in place, but as we'll see, that filum terminale is not the only thing protecting the spinal cord. So that little bit of looseness to your spinal cord dramatically outweighs any potential risk that you get from that pulling down of the spinal cord and causing the pain and the weakness and the loss of sensation and all of those other things that can go along with that.
Question.
Yes.
But if you cut it, isn't that like the nerves as well? Don't you lose some feelings or something like that, or anything else going on, or is it just holding the spinal cord in place?
It is one of the things that helps to hold the spinal cord in place, but again, there are other protections around the spinal cord as well. So
Okay.
the loss of this one protective coating, again, in simplest terms, if you've got a very valuable vase that you're sending to grandma, you know, one of the things that you may do is you're going to wrap it in bubble wrap, you're going to put a bunch of peanuts around it, and you're going to tape it in place. If you cut one end of the tape that was holding it in place, it's not as stable inside that box, but you still have the bubble wrap, you still have the packing peanuts, you still have the tape holding the top part of it. So it's still going to be in place, and it's still going to be, it's still going to be stable, and it's still going to be safe. And like I said, it definitely is less of a concern than the severe permanent damage that could occur to these nerves by forcing them down into a position that they're not supposed to be in. All right. Okay. So, as I mentioned, we have a spinal cord. And I'm going to cheat and draw it this way. And that spinal cord is actually divided into 31 individual segments. These 31 individual segments, as we can see here, are given names much like the vertebrae are, with cervical, thoracic, lumbar, sacral, and coccygeal. Coccyx. Excellent. Now, remind me again, when we talked in terms of bones, how many cervical vertebrae did we have again? 7. How many thoracic vertebrae did we have again? 12. How many lumbar vertebrae did we have again? 5. How many vertebrae fuse together to form the sacrum? 5. 5 for the sacrum. But how many fuse together to form the coccyx? 4 for the coccyx, right? However, remember, the coccyx is pretty tiny. Probably not going to associate 4 nerves to that. So really, those fuse together so early, and it's such a tiny thing, it really just counts as one bone. If we do the math and add all that together, what does that equal? How many bones do we have there? 30, excellent. Well, each segment of the spinal cord actually has a pair of nerves that come off of them. So each segment has a pair of nerves that come off, one to the left and one to the right. And so we have 31 segments of the spinal cord, and therefore we have 31 nerves. Or let's be more specific, spinal nerves. And that numbering is almost perfect, but a little bit off. And the way that works, as we see nicely from the illustration here, is that we actually have 8. So let's actually cheat. When we talk about the nerves, we have 8 cervical nerves, and then 12 thoracic, and 5 lumbar, and 5 sacral, and 1 coccygeal. And that, of course, gives us our total of 31. Typically, the way we identify a spinal nerve is by the vertebrae it travels under. So if you notice, for instance, if we come over here to our illustration, T9 comes out underneath thoracic 9 vertebrae. L3 comes out underneath the 3rd lumbar vertebrae. However, notice we've got a different number of cervical and a different number of nerves versus the bones. And that's because what happens with C1, our first cervical nerve, is, as you can see, it comes up above C1. And what was C1 called again? Well, I knew learn the name of C1. Atlas, excellent, right? Atlas, perfect. And then C2 comes above the axis, and C3 comes above. And so notice all of these, C7 comes above. And that means here underneath C7, we have that 8th nerve. So because our cervical go above, we get an extra cervical nerve. And so 31 nerves with only 30 bones is how the math of that works out. All right. And these nerves go to different parts of our body, as we will see. So again, 8 cervical, then 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal. Now notice, as we talked about, these nerves, at first, C1 goes up above, and then the rest go below, as we see here. But because the spinal cord stops before we reach the end of the vertebral column, that is why these nerves have to travel further and further until they can get to a point that leaves, and that is where we get that cauda equina, that horse's tail from those long bundles of axons going down there. Now I want to cheat because I don't have the picture here, and I'm going to go back to it actually I'll go back to it in a second. You might not have been aware of any of these things, but you kind of were. Because, all right, you or someone who loved you very much gave birth to you, or you gave birth to somebody else, all right? If you've given birth, what is one of the big questions you get asked when you get wheeled into the hospital?
Do you want an epidural?
Exactly, absolutely. That is the big question. Do you want that epidural? What is
Hell
an
yeah.
epidural?
Um, it's pretty much a source of medication that they insert a needle into your epidural zone in your spinal column,
Mm-hmm.
and it pretty much numbs your entire lower half of your body. And for me, it numbed about a little above my belly button and all the way down my legs for about 14 hours.
Excellent. As we talked about, right, what that medicine actually does is it blocks the voltage gated sodium channels. So as we talked about, that signal, not just the pain coming up, but also motor control going down, tactile sensation coming up. None of those things are, all of those things are blocked. And where did they make the insertion of that needle, with that needle?
It was right around from, well, I mean, I was in active labor. So
Right.
from what I could remember, it's like right at my, like, I want to say L1 and L2 area.
Close. Anyone else want to have? Actually, it's typically lower. Typically, where they are going to give that epidural is somewhere between the L3 and L5 region of the vertebral column. All right, because notice the goal is to take a needle and put that needle in the epidural space. We'll talk about what the epidural space is in just a second. But that epidural space, as the name might indicate, is above the epidermis, before the nervous tissue. However, when you have a very late term pregnant female who's in a fair amount of pain, right? They may wiggle a little bit. They may, you may be a little nervous. There is the potential that the needle could go in a little too far. And the advantage of being down here in the L3, L4, L5 region of the vertebral column is you are inferior to the spinal cord, and you are not going to hit the spinal cord, and you are not going to give damage. So the reason they give that epidural so low, yes, is because they want to anesthetize the lower half of the body, but it's also so that they are not risking damage to the spinal cord. So that is why they're doing it in that inferior location.
I actually have a question about that because so my friend, she gave birth at a teaching hospital and they messed up her epidural to where after they had taken it out, she had a like buildup of cerebral spinal fluid like in her neck, and they pretty much had to like open it and create like a blood patch. Like what exactly happens for that to be happening?
That's a great question. I'll tell you what. Let's table that question for just a couple of minutes. I want to finish with the spinal cord because when we talk about the protections, we will actually see the epidural space, and we can talk about that, and then we can talk about what happens when it goes bad.
Okay.
Okay, but let's actually look at the anatomy of that, and then when we talk about it, it will make more sense when we see it. Okay, so we got a couple more slides before we get to there, but I promise you we'll get to there, and I promise you we'll answer that question. All right, because it's an excellent question and definitely one that I wanted to address. All right, but like I said, give me 2 more seconds and we will get there. All right, the last thing that I think this is the last thing, and then we'll get to that. Almost the last thing. 2 more things before we get to that. Is that when we talk about these nerves that come off, these bundles of axons, and again, remember they're leaving the spinal, they're leaving the central nervous system. So they are bundles of axons outside the central nervous system, and so of course we call them a nerve. They contain both sensory and motor information. So what that means is if I cut a nerve going to my hand, not only do I not feel anything with that finger anymore, but I can't move my finger anymore as well. So all spinal nerves are a mix of both sensory and motor, both afferent and efferent information. This is important because when we get to our cranial nerves, the 12 cranial nerves that we're going to talk about possibly today, those aren't. Some are sensory only, some are motor only, and some are both, or mixed nerves. But all of our spinal nerves are mixed. So if you cut a spinal nerve, you lose tactile sensation and the ability to move it. All right. Let's do that and then do that. If we take a closer look at a nerve, what we see is it isn't just a random collection of axons. It's actually very, very organized. In fact, this organization may seem slightly familiar. Notice here, I have my myelinated axon. Now, of course, this is in the central nervous system, so what cell is forming that myelin again? And the central nervous system, it's not the oligodendrocytes, it is the easier one to spell, which is the Schwann cells. Excellent. But notice if you look closely, you see that this myelinated axon and all of the myelinated axons have an areolar connective tissue that wraps around each one of them. Almost like it was insulating them and isolating them from each other. All of these axons, long cylinder, almost fiber-like structures, are then bundled together into a bundle. And they're made into a bundle by a dense, irregular connective tissue. This bundle and 14 of his closest friends are then bundled together along with blood vessels to form our nerve. Oops, nope, I don't want that. Pink. So all of our bundles and blood vessels are bundled together into the nerve by a dense, irregular connective tissue. What does all of this seem very similar to? Where has this type of anatomy sound familiar to us from? From muscles, absolutely. Right? So perfect. Let's erase that. Guess what, based on its location, we call this areolar connective tissue that wraps around individual cells. Well, right, it was an endomysium in muscles, but this is around a nerve, so it is an endoneurium. Guess what we call the bundle? Well, you're right. The dense irregular connective tissue that forms the bundle is going to be based on its location called a perineurium. But what do you think we call the bundle? Excellent. Perfect. And indeed, it is a perineurium. And the dense irregular connective tissue that we use to form the nerve itself is indeed the epineurium. Excellent. So notice we learned this anatomy once, we get to use it twice. A nerve is formed by a bundle of fascicles and blood vessels wrapped together by a dense irregular connective tissue based on its location called an epineurium. A fascicle is a bundle of axons made by dense irregular connective tissue, which based on its location we call a perineurium. And each individual axon, myelinated axon, so notice the axon is inside of it, the myelin is inside of it. There's an areolar connective tissue that wraps around the myelin and the axon, and that areolar connective tissue based on its location is called an endoneurium. Now, in grading the muscle lab exams, there were several instances where I would have an arrow just like this green one and ask you to identify that structure, and someone said dense irregular connective tissue. Or I would say identify this tissue based on its location, and someone would say fascicle. Or I would say identify this tissue type, and someone would say perineurium. Right? Notice all of those answers show knowledge, show an understanding. But the problem is, did you answer the question I asked? No. This here is the structure. So if I ask for a structure, your answer needs to be fascicle. If I ask for a tissue type, then this needs to be dense irregular connective tissue. And I ask for the tissue, oops, based on location, then that is the perineurium. So again, many of you showed knowledge on this when we were doing this with the muscles, but lost points. As I've said many times, people lose points not because they don't know the information, but because they don't read the questions carefully. Please read the questions carefully so you can answer them correctly. All right? But the good news is this is something that hopefully should be fairly familiar. And again, here is the anatomy of all of that we see here. If you don't like my drawing, then here are the drawings showing all of those things that way. All right, questions on that? Oh, and here's another pretty picture that shows this. And you need to be able to recognize this histologically, which is on the histology review. So I know that is on there as well. All right, excellent. Now let's get to Madison's question. Here we see the protective coverings of the spinal cord. And I'm going to cheat and actually draw this one. So the first thing we need to do is, again, and I'm going to totally cheat with this drawing here. Actually, hold on, let's make it black. Here is my body of my vertebrae. Here is one pedicle. Here is the other pedicle. Here is the lamina. And then let's go ahead and put a spinous process on it and a transverse process on it. There you go. My beautiful, real-life looking vertebrae. And then, of course, smack dab here in the center is our beautiful butterfly shape, oval shape spinal cord. Now, the good news is we are still going to have those 3 layers of meninges. There we go. Still have those same 3 layers of meninges. A dura mater, and it's still going to be a double layer of connective tissue. An arachnoid mater, that is still going to form a subarachnoid space, and our shrink wrap pia mater. So all of those are still going to be there. But there is some big differences. And one of the big differences between what protects our brain and what protects our spinal cord are the bones that make them up. The bones that protect our brain are the skull bones. And skull bones are held together by sutures, and they do not move. They do not change shape. Our vertebral column is made up of all these individual vertebrae, and all these individual vertebrae need to be able to move in relationship to each other. So our spinal cord is contained within bones that need to move, where our brain isn't. So the first big difference between these is if my vertebral column needs to be able to twist and turn. Do I want to attach my protective coating, my dura mater, directly to the bone the same way we do in the skull? No. So we are going to need a dura mater. And let's go ahead and make that dura mater brown. We are going to need, that's not brown. That's brown. We are still going to need a dura mater that surrounds this. and it is still going to be a double layer of material that surrounds it. But this time, the superficial layer does not connect to the bone. The bone does still, and I guess we can draw it here in yellow. The bone is still going to have a periosteum. We know, like all bones, it is still going to have a periosteum. That periosteum just isn't a part of the meninges here in the vertebral column, where it is in the brain. And so notice what we get here is a space between the periosteum and the dura mater. There is no space, not even a potential space up here, because it connects directly to the bone. But here there is actually a space in between, and not surprisingly, the space here in between the periosteum and the dura mater, or really, you can think of it as the space above the dura mater, is the epidural space. So when you are getting that epidural, notice the goal is to place the medicine right here in this space, not touching the spinal cord, not even going within the protective layers of the spinal cord. So we're not going to protect the spinal cord. And again, remember, at that level, it's not even the spinal cord anyway, it's just the axons. But we don't want to enter the protective coating. We just want it in this space. Now, of course, this space is more than just there for medicine when you're giving birth. It is going to contain areolar and adipose connective tissues in that space. That is an actual space between the periosteum and the dura mater. Okay? So far, so good? All right. We also then are going to have we'll use red for this one. Perfect. We are also going to have our arachnoid matter. So let's actually cheat and do this. So arachnoid matter is that one. Our dura mater is that one. Now, as we know, there is going to be a subarachnoid space. And that subarachnoid space contains, and I'm just going to abbreviate it here for space, cerebral spinal fluid. Okay, we're comfortable with that. But notice also, as we talked about last time, there is a space between the dura and the arachnoid matter. In our brain, it is a potential space, and that potential space we call the subdural space. All right, that's where we got that subdural hematoma. It was a potential space. Here in our spinal cord, it isn't a potential space, it's an actual space. And this actual space contains not cerebral spinal fluid, because that's what's in the subarachnoid space, but instead it contains interstitial fluid. So again, notice rather than having the dura mater and the arachnoid mater smashed up against each other, there's a little space in between. And again, this fluid-filled space allows for more flexibility, more movement because that our spinal cord needs to be able to withstand that movement that's taking place in the vertebral column, and our brain doesn't have to worry about that. The rest of the anatomy, of course, is absolutely the same. Our subarachnoid space contains cerebral spinal fluid. Oops, no, not what I wanted. Blue. Our pia mater is that shrink wrap on the outer surface of the tissue, stabilizing it, holding the blood vessels in place. So we still get that nice pia mater. That's the same. So really, the big differences, oops, no, didn't want to do that. The two big differences that we have here are that the subdural space is an actual space that contains interstitial fluid, and the epidural space, which is above the dura mater, that contains areolar and adipose connective tissues. So the arrangement of the tissues are the same. There's just two additional actual spaces that are containing stuff. All right, so now, as was mentioned, what can sometimes occur, and what color should we use for this? I haven't used purple yet, so let's stick with purple. The goal of an epidural is to bring that needle, and obviously not bring it in from this direction, but there's less stuff down here, so it works. To insert that medicine into the epidural space, where it can then diffuse across all these protective membranes, and again, help to paralyze, right? Stop the flow of action potentials and stop the spread of that signal. Unfortunately, what can sometimes happen is sometimes they insert it too far. If they insert it too far, they damage those tissues, and several things can occur. One of the things that can happen is that it can actually cause some of the cerebral spinal fluid to leak. If the cerebral spinal fluid leaks out of that space, several things can happen. But if you think about it, one of the problems that can occur from that is that you are going to lose some of the buoyancy of your brain, because remember that fluid is holding it in space, and so it can start to sag a little bit in the cranial cavity. That can cause incredibly painful migraine-like headaches that can last from that draining of the cerebral spinal fluid out of that space. So that can cause some major issues and problems that way. The other thing that can happen, and I think this is what happened when Madison was talking about, is in damaging those tissues, you can irritate those tissues. And as we know, when you irritate tissues, that can cause inflammation, that can cause swelling, that can cause edema to occur in those places, and that can increase the fluid pressure in the subarachnoid space, and that can cause potential problems as well. So my guess is that's probably what was happening if they had to drain something in the neck region, is probably they'd irritated the tissue and they were getting edema in that space because of damage.
Well, yeah, and she also said that she like couldn't get out of bed for like 5 days. Like she couldn't move her body at all.
Wow.
So they obviously did something very, very wrong. Whereas mine, I was just really, really numb. I think I got a little bit of an overdose because my anesthesiologist was like, "Yeah, you're 5'6", right?" And I like looked at him and I was like, 5'3". He just walked away, gave me a thumbs up. I'm gonna feel great, and I was the most numb lady in labor on that day. It's great.
Yeah. Well, there are some advantages to that. My wife also had an epidural with hers, and especially for her first bout, she was in labor for a very long time, and so both my mom and her mom were in the room with her, where she had, they have the monitor set up where you can actually watch the contractions, and they're like, "Ooh, look, there's another one," and she's like, "Oh man, if I wasn't anesthetized right now, I'd be beating them."
Oh yeah.
They were all getting all excited watching it, and she was. But
yeah, like my dad was astonished because my mom didn't do an epidural at all with any of us, and so to watch it, he's just like, "That's kind of nice. Like you get to enjoy it a little bit." And I was like, "Yeah," but beforehand it was awful. I would definitely recommend getting an epidural if that's what you want to do. But my contractions were so close together, I didn't have any time to prepare for them. They were about 15 seconds apart. So the epidural definitely
then that, you know, that might have been the other reason why they, and again, I'm literally armchair quarterbacking this, obviously, so again, all of my examples that I'm using are theoretical in this case, but theoretically what happens sometimes if someone is transversing through labor more rapidly, then one of the things they will do is they will do a larger bolus to try to get a faster effect, because otherwise the baby could be born before the medicine is fully kicked in. So that might have been the other reason. If they were worried that you were progressing too rapidly, he might have increased the bolus because of that as well. But again, I'm merely speculating.
No, that's probably right too, but I didn't care. It was great.
All right, excellent. So again, I've done an amazing job of recreating this with the illustration here, but let's look at the pretty picture from your textbook, and we see all of these regions as well. So again, notice here we have that epidural space containing the adipose, containing the areolar connective tissue. From there, we then have the 2 layers of dura mater that do not connect to the bone. Notice from there, we then have the subdural space, an actual space between the dura and the arachnoid matter. And as I mentioned, it contains interstitial fluid. So the space between the dura mater and the arachnoid matter is an actual space here, filled with interstitial fluid. Then we have that arachnoid matter. Inside of that, our subarachnoid space, which contains the cerebrospinal fluid. And then there is that shrink-wrapped pia mater on the outer surface, stabilizing the blood vessels and stabilizing the axons that are coming out of the spinal cord as well. All righty. Questions on that? All right, now that we have done the gross anatomy of the spinal cord, we need to do the microscopic anatomy of the spinal cord. And looking at the time, I'm not sure we're going to get to the cranial nerves today, so we may have to bump them to tomorrow. So let's do that. But let's go ahead. We still have a fair amount. I know I went a little long on this. It is 11 o'clock. But we still have a fair amount to do. So let's see if we can at least get through the anatomy, because one of the important things we're going to finally be talking about with the spinal cord is, again, emphasizing this relationship between location, functional classification, and structural classification. So we need to start doing some of that. So we need to talk about the microscopic anatomy of the spinal cord. So it's 11 o'clock now. Let's take a 15-minute break and return at 11:15. And we will start the recording again then. All right, any questions before we take our break? All right, go caffeinate again so that we can make it through the last stretch. See you guys in 15 minutes. Any questions before we get started? All right, then let's talk about the anatomy of the spinal cord. We have a nice picture of it here, but again, I think this is one of those times where actually taking a few moments to draw it, I think is definitely something that can be beneficial. Our spinal cord is a big kind of oval shaped structure. Actually, hold on, let me move that to there and that down there, and that'll give me a little more room to play with. Excellent. Something like that. As we know, there is a small space in the center of it. Oops, there. And that space in the central is the central canal. Hopefully, as you can know, that central canal is contains what substance? Substance is contained in the central canal of the spinal cord. My mic is on, you guys can hear me, right? There you go, cerebrospinal fluid, excellent. Let's ask a trigger question. It is lined on the inner surface by a single layer of cells. What cells do you think line the space of the central canal? Ependymal cells, excellent. Much like the cerebrum, much like the cerebellum, the spinal cord is divided into 2 hemispheres, a left and a right. And so they have invaginations. Oops, nope, don't want that to be purple. They have invaginations, one posteriorly and one anteriorly. Both of these are invaginations on the midline. However, the one on the back is smaller, so it is the posterior median. Oh, that's way too big. Posterior median sulcus. On the posterior side, one of the ways we can tell the front from the back. And the other one is obviously anterior, still median. But this is a much larger invagination, so it is fissure, anterior median fissure. There is another way we can tell the orientation, anterior versus posterior, of the spinal cord, and that is in the organization of the white matter and the gray matter. Unlike the cerebellum, unlike the cerebrum, with our spinal cord, the gray matter is deep, and the white matter is superficial. Right? It is different. This is different from what we saw in the cerebrum. This is different what we saw from the cerebellum. So the gray matter is deeper, and the gray matter isn't evenly distributed. It has kind of a butterfly-like appearance to it, where it has an enlargement here, and it has an enlargement here. And then the largest bit of gray matter is towards the front. So we have these three enlargements that are asymmetrical in their size. And we identify these as gray horns. So again, I think I may need to make this, let's see if this works. This is our anterior gray horn. This is our lateral gray horn. And then this one up here then is our posterior. Oops. Horn. Now, I've used the terms anterior and posterior for these. But remember, those terms are interchangeable with dorsal and ventral. Anterior and posterior for the median sulci, median fissure, the gray horns, tend to be the terms that are more commonly used. But there's absolutely nothing wrong with calling it the ventral gray horn or the, the ventral gray horn or the dorsal gray horn. That's just by convention. And again, by convention, it's the rules that most people tend to use. These are the ones they use. But again, remember, these terms are interchangeable for us. Not only are there enlargements to the gray matter, but there are enlargements to the white matter as well, what we call the white columns. And so not surprisingly, this is the posterior white column. This is the lateral white column. And this is the anterior white column. Excellent. We have this symmetry left and right. I know my drawing's not symmetrical, but it's close enough. These differences anterior and posterior. And notice, though, they do connect a little bit in the middle. This little bit of gray matter that connects them in the middle. If you remember when we talked about having axons that connected across the middle, we called them commissural axons. Well, this is gray matter that connects in the middle. So this is actually referred to as the gray commissure. The little bit of gray matter that crosses the midline and surrounds the central canal. And I guess we should write central canal here. And then in parentheses, the ependymal cells. Oops, hold on. There you go. Nope, sorry. Why do I keep doing that? Epidermal cells. There we go. I'd outline that there. So we have that anatomy there. All right. So these are the anatomical features. Of course, as we know, gray matter is where the cell bodies and, of course, their dendrites are located. And then the white matter is, of course, made up of axons, primarily myelinated axons. So when we talk about these different locations, posterior gray horn, lateral gray horn, and anterior gray horn, these are where our cell bodies are going to be located. So these are our cell bodies in the gray matter. And as we know in the nervous tissue, not only do we have cell bodies in different locations, but different locations have different functions. It turns out the neurons that are located here in the anterior gray horn, and let's start adding some color to this now. These are where we find the somatic motor neurons. Somatic motor neurons are located here in the anterior gray horn. And actually, I'm going to cheat and put this up here, right there at the top of my anterior gray horn. So now we have a location, we have a function, and now that we have a function, do we also know the structural classification? So this is our functional classification. It is a somatic motor neuron. What would its structural classification be? It's a motor neuron. What must its structural classification be? Get used to these questions. There's going to be a lot of them on the exam and all of them on the reviews. What would the structural classification of a neuron whose cell body is located in the anterior gray horn? It is going to be multipolar, absolutely. It's a multipolar neuron. So here, inside of our anterior gray horn, we have a cell body with dendrites coming out of it. And its axon is going to leave the spinal cord and travel out to the body. When it leaves the spinal cord and travels out of the body, as it leaves, it forms a structure that is known as the ventral root. That ventral root, not surprisingly, is going to meet up with a dorsal root coming down. And when they meet up, they are going to come together. So let's see if I can sneak that name in here. They are going to meet up and form the spinal nerve. So remember, we had talked about how each segment of the spinal cord has a pair of spinal nerves coming out of it. The same thing is clearly happening on the other side as well. So here we have a location, anterior gray horn. That location has a specific function, somatic motor neurons. Those neurons have a specific type, multipolar. So notice location, function, structure, all go hand in hand in hand for that particular neuron. Excellent. This is somatic motor. It turns out our posterior gray horn. So questions on that one before we move to the next one. Stunned silence. I love it. Excellent. Our posterior gray horn is responsible for all sensory input. It receives all sensory input. I need to make that smaller now. All sensory input comes into our posterior gray horn. Now, Sensory. What is the structural classification of a sensory neuron? Most sensory neurons. Unipolar, excellent. So if we have a sensory neuron, that sensory neuron by function, its functional classification is sensory. Its structural classification is going to be unipolar. And what we're going to need is a location for those neurons. And it turns out the location for those neurons is not actually in the spinal cord. It is in a collection of cell bodies located outside of the central nervous system in a structure known as the dorsal root ganglion. And again, you could call it the posterior root ganglion. That is fine as well. But what we get here is a cell body that has one process coming off of it, and that one process is an axon. That axon comes in the spinal nerve, up the dorsal root, and then ultimately leads into the posterior gray horn. So out here on the dorsal root is an enlargement, and this enlargement on the dorsal root is the dorsal root ganglion. And that dorsal root ganglion is going to be the third way you can tell the front of the spinal cord from the back. The front has the larger fissure, the front has the larger anterior gray horn, and the back side, the posterior side, is where you find connected the dorsal root ganglion. There is no ganglion on the ventral root. There is a ganglion on the dorsal root. So if you find the dorsal root connects to the dorsal or posterior part of the spinal cord. Now, notice one other thing about this. Notice that our dorsal root only contains sensory axons. Make that 14. Let's make that red. It only contains sensory axons. Whereas notice our spinal nerve is mixed. Both sensory and motor. Whereas our ventral root, notice, is motor axons only. All righty. Questions on that? Notice we still have another gray horn. So we still have more gray matter where we can have more axons. But we've done all the sensory, we've done all the somatic motor. What could possibly be left?
Is it the gray commissure? Or?
No, well, gray commissure is primarily interneurons carrying information in between. Interneurons are good. But notice here I said all sensory, but down here I didn't say all motor, I just said somatic motor. Is there any other type of motor? Well, I'm asking the question, so what's the obvious answer? The obvious answer is yes. What is the other type of motor we might have? Autonomic, exactly. And that's with the lateral gray horn. The lateral gray horn is where we find the cell bodies. That functional classification is our autonomic motor neurons. And of course, if these are autonomic motor neurons, what must their structural classification be? Multipolar, excellent. Excellent. So here in the lateral gray horn, we are going to have a cell body. Hold on. Let me sneak this up here and do that so we can see that a little nicer. Cell body here. It is a multipolar neuron, so it is going to need dendrites coming off of it. It's also going to have an axon that comes off of it. And which way do you think this axon is going to want to go? Is it going to want to go out the dorsal route, which only contains sensory axons, or do you think it's going to want to go out the ventral route that only contains motor axons? Not your question. It's a motor neuron. It's going to want to go out the ventral route. Exactly. Going out to our spinal nerve. Now, let's take this one step further. Now, obviously, like I said, all sensory comes in. Special sensory, somatosensory, visceral sensory, all sensory comes into the posterior gray horn. But here, our motor is divided. With somatic motor, what is the effector that it is? Let's wait, hold on. Need to make that blue. What is the effector that somatic motor neurons communicate with? What is the target of a somatic motor neuron? What tissue does a somatic motor neuron terminate and release its neurotransmitter at? Exactly, skeletal muscle. Whereas up here, what is the effector or effectors of our autonomic motor neurons? One week till the exam. What are the effectors of our autonomic nervous system? Glands. What else? Smooth muscle and cardiac muscle. Excellent. Perfect. So there you go. Location, function, structure, all go hand in hand in hand. If I give you one of these, you should be able to figure out the other three. And notice one more thing. Notice when we talked about that spinal nerve, if I cut that spinal nerve going to my finger, notice we cut all three types of axons. So yes, as I mentioned, you don't feel anything on your finger anymore. You lose your voluntary control of your finger. But notice you also lose your autonomic control. Your finger won't sweat anymore. Your erector pili muscles won't make your hair stand up on end anymore. You won't be able to dilate and constrict the blood vessels in that finger anymore. You lose all control of all motor and all sensory going back because that spinal nerve contains all of those things. All right? Questions on that? All right, I've done, quite frankly, an amazing job of drawing this. But let's look at the pretty picture from the textbook. Notice here, we see our invaginations, and notice they haven't done as good of a job of differentiating the ventral median fissure from the posterior median sulcus. The posterior actually looks a little longer to me, at least in the way the illustration has been drawn here. So that's not a good indication. And you might even argue that our anterior gray horn is kind of similar in size to the posterior gray horn. Our lateral gray horn is a little sad, but notice we at least still have that dorsal root ganglion out there to make sure we know this is the back and this is the front. So we look at that here. Here, we see the nice pretty picture from your textbook that does a little better job of emphasizing this. Again, notice our three enlargements: posterior gray horn, lateral gray horn, anterior gray horn. The anterior great horn is the largest. Notice here that anterior median fissure is much bigger than that posterior median sulcus now. They've done that nicely. And we still have our dorsal root ganglion connected to the back. Now notice, again, be careful. Look at this picture here. Often when this is on the slide, notice this is towards the front. So you may see this dorsal root ganglion lying up here, but what you have to do is follow its axons back. Its axons go to that. So this is still the dorsal root ganglion, even though it's tilted a little bit to the side. You see that sometimes on the microscope slides. When they mount it, those dorsal root ganglions kind of dangle down. But you follow, it's not where it's located in relation to the spinal cord. It's actually how it connects to the spinal cord. So we basically have three important locations here where we find cell bodies: the anterior gray horn, which is where we find our somatic motor neurons that are multipolar; our lateral gray horn, where we have our multipolar autonomic motor neurons controlling smooth muscle, cardiac muscle, and glands. Notice, as I mentioned, both somatic, special sensory, visceral, all sensory information comes into that posterior gray horn. We're not going to worry about differentiating the regions, because what we care about is where its cell body is located. And its cell body is located out here in the dorsal root ganglion. And this is sensory, so it's unipolar. This is motor, so it's multipolar. This is motor, so it is multipolar. So we have locations, we have functional classifications, and structural classifications. All right. Questions on that? Now, again, white matter isn't just random bundles of axons. We saw how the nerve is very organized. These white columns are equally organized. You don't need to know the anatomical names of any of these. You don't need to know where the reticular spinal tract goes to or comes from or anything like that. You don't even have to distinguish the ascending from the descending. But remember, as we mentioned, ascending is sensory coming up, descending is motor coming down. The only reason I show you this is because, again, I just want to emphasize that not only is the gray matter precisely organized, but the white matter is precisely organized as well. But none of the anatomy of this, other than the columns. You need to know the posterior white column, the lateral white column, and the anterior white column. Other than knowing the columns, you don't need to know any other anatomy for the white matter of this. All right. And again, here you see kind of the illustration that we were talking about, that dorsal or posterior root is the sensory coming in. And that's also where the dorsal root ganglion is located for unipolar neurons. Our ventral root is motor only, carrying both the somatic and the visceral motor, carrying that information out. And then again, it comes to the nerve. And once it comes to the nerve, it actually branches not once, not twice, but 4 times. So let's actually sneak back to our picture and see if I can sneak that in. Perfect. I can cheat and do it on this side. So let me do this first. Where do I need this? Let's put this, this doesn't fit anywhere. We'll put it up here. So I'm just going to draw them as lines this time because it is easier for me. So, but let's make the lines at least a little bigger. And we'll make them black. So we have that dorsal root coming out. We have the ventral root coming out, and then they come together and meet. On our dorsal root, we have our dorsal root ganglion. Oh, see, now I've done this. Hold on. I got to undo all that. All right. I'm going to cheat and put it a little bit closer. So our dorsal root will have come out like this. Our ventral root will have come out like this. We'll make the dorsal root ganglion a little smaller to form our spinal nerve. Whoops. To form our spinal nerve. Okay? When that spinal nerve comes out, it branches not once, not twice, not 3 times, but 4 times. There are 4 big branches that come off of that spinal nerve. So I'm going to cheat and make this small and just put SN here for the spinal nerve. And then when the spinal nerve branches, it basically forms 4 branches or 4 arms. And if you remember back to your bones, one of the names we used for a feature that was arm-like was a ramus. So what we have here are 4 different rami that branch off. The first ramus is the dorsal ramus. Dorsal ramus goes to the posterior part of the body, goes to the back part of the body, and basically innervates the skin and muscles of the back. So that is our dorsal ramus. Our second ramus is the ventral ramus. It is the largest of all of the rami. It innervates the anterior part of the body and the limbs. So it goes to the arms and legs, and it goes to the front of the body. It is the largest of all the rami. These other 2 rami collectively, can you see that if I write that there? Okay. Collectively, they are known as the rami. Rami is the plural of ramus. Rami communicantes, which is incredibly fun to say. But what I will tell you is they each do have their own individual name. The lateral one is what is known as the white ramus. And the medial one is what is known as the gray ramus. So the medial one is the, and let's actually put, I can't put parentheses around lateral anymore, but we can at least do it here. The medial one is the gray ramus, because I don't want you to call it the medial and lateral. So let's actually go ahead and delete that one and write it again. And the lateral one, I'm only calling them lateral and medial just so you can tell which one I'm talking about. You need to use the correct names for them: white ramus and gray ramus. So again, the lateral and medial is just for orientation purposes. And these involve our autonomic nervous system. You are going to learn everything you wanted to know and more about those next week on Monday and Tuesday when we talk about the autonomic nervous system. So again, here I have this, oh no, wrong button. I meant to save that, not delete it. But if we go back to the pretty pictures from your textbook, we can see here, here we see the nice pretty picture. And notice here when we look at the nice pretty picture, where's my annotation? There it is, highlighter. Here is the dorsal ramus coming to the muscles and the skin on the back. Here is the ventral ramus, in this case just going to the front of the trunk, but would also go to the arms and the legs. And then here are the 2 rami communicantes, the white ramus and the gray ramus, and they play a role in our autonomic nervous system. And so we'll talk a lot more about those next week. All right, so dorsal ramus, skin and muscle of the back; ventral ramus, anterior trunk and limbs; the gray and white rami, the rami communicantes, and here we see them coming off here as well, play a role in the anatomy of our autonomic nervous system, which you definitely should spend some time this weekend looking at. If you read the chapter on the autonomic nervous system, you will see that there is a ton of information in there. So as always, obviously the things that I talk about in lecture are the things that are most important. Those are the things that you're going to be tested on. But that is absolutely, positively, definitely true here for this section. For studying the autonomic nervous system, which I encourage you to get a head start on during this weekend, make sure you are looking at the lecture slides. Look at the lecture slides first to see what we're going to talk about, and then go to the textbook. Don't just read the entire chapter of the textbook. There's way too much information in there. Focus on just the things we're going to be talking about on those 2 days, because those are the things that you're going to be responsible for on the exam. All right. And one last pretty picture. Notice here we see the sensory. Again, some visceral sensory comes up through those rami communicantes and through the ventral and dorsal ramus to the spinal nerve, to the dorsal root, and they're unipolar, and their cell bodies are located in the dorsal root ganglia, and their axons come into the posterior gray horn. Oops, where's the other one? Ah, here we go. And then here is the motor, autonomic motor, somatic motor. Their axons come out, multipolar neurons located in the gray horns inside the spinal cord. Axons come out the ventral root to the spinal nerve, and then off from there. But notice our autonomic go down the rami into the sympathetic pathway that we'll talk about next week. All right, how are we on time? One more thing, excellent. All right, so yeah, we can do this. So the last thing that I want to say today, and this is where we will pick up tomorrow, is to talk about the spinal nerves. Those spinal nerves, as we mentioned, they come out right away. The dorsal and ventral the dorsal and ventral roots come together to form the spinal nerve. But then remember, it branches right away to form the dorsal, to form the gray and white, and to form the largest, which are the ventral branches. And as you can see from this illustration, most of those ventral branches, there are a few that just pretty much go out to one location and stop. Most of them branch to form these elaborate networks of nerves. And these elaborate networks of nerves are called the plexes. You have 4 major plexes in your spinal nerves. For those, we are going to identify all 4 of the plexes. We are going to identify which spinal cord roots, which spinal nerves come together to form them. And for each of the 4 plexes, we'll identify one or a couple nerves that are important, and we'll talk about why they're important as well. All right, that's going to be some more dense information. Basically, from now till the end of the section, these next 3 lectures are all going to be dense information. So I think I've talked for a lot. It's a little bit early, but we have caught through. Again, we didn't get to the cranial nerves, but there's not enough time to stop that. I don't want to get into this and have to stop midway. So I think this is a good natural stopping point. So you guys get a teeny bit of a break today. But that means we got a little bit more to make sure we get done tomorrow to make sure that we get it all in. Because like I said, I definitely want to make sure we get through our cranial nerves before we start, before the weekend. So in fact, even though we're here at the plexus, I think maybe what we'll do is we'll actually start tomorrow. So this will be the game plan. After you take your 10-point daily quiz tomorrow morning, and again, I will make it available at 7:50, but I'm still going to end it at 8:10. So you'll have 10 minutes for the 10 questions, but I still want to start lecture at 8:10 because I don't want to go too much later than that. So I'll start making it available a little bit early. Maybe I'll make it available at 7:45. So between 7:45 and 8:10, you'll have a chance to take your 10-point daily quiz. And then at 8:10, what we'll do is we'll actually start with the cranial nerves. So make sure you have that cranial nerve handout printed out, or at least your own graph or something that you want to fill out as we go through that information. All right? Questions on any of that?
Yeah, I got a question.
Yes.
On when we're talking about like location, structure, function, is there, besides taking the notes, is there like a slide or something where we can go to just review those?
Well, I mean, your textbook obviously talks about these things. I'm sure I emphasized it a lot when I was doing the histology review. So if you haven't looked at that histology review yet, look at that. And so, yeah, obviously we're going to talk about it in lecture. It's in the handouts and it's in your textbook. So, I mean, those are the same resources for all the information.
Okay.
Okay. All righty. Great question. Any others? All righty. Then I will see you guys bright and early tomorrow morning. Have a good day. Stay healthy, stay safe, and I will see you tomorrow.